Welcome everyone. In this lecture, we are going to complete a chapter which is in 12th NCERT, chapter number 11. It is Biotechnology, Principles and Processes. Now, wherever technological aspect comes from there, for NEET, the weightage is more. So, try to understand the concepts very clearly. And again, we are going to complete the whole chapter in one shot. So, who coined the term biotechnology? Karl Erke is the scientist who coined the term biotechnology. And who is father of biotechnology? Paul Berg is father of biotechnology, right? So I have taken some historical account related to biotechnology. We will see that historical scientific details and then we will get into the chapter, right? So here there is a scientist called René Descartes, right? Who is he? He is a French philosopher. He is a mathematician and he is a biologist also of 17th century. Now he told that all human knowledge, especially natural sciences, whatever we know related to natural sciences, were directed to the developing technologies. So we are using all our natural science knowledge to develop technologies which will benefit us only. So we are not bothered whether it's benefiting a plant or an animal or a microorganism or an ecosystem. We are just seeing whether it is benefit for us, then we will propagate it, we will multiply it. So that's what he felt, that in 17th century, this scientist, who is a French philosopher, mathematician and biologist, so he thought that all the knowledge related to natural sciences has directed to develop the technologies, and these technologies were helping to give more comforts to the man life, were giving more comforts, more values to the man life. That means we can simply tell in this sentence that the whole approach of understanding biology, the whole approach of understanding the natural processes became anthropocentric. Man-centric is called anthropocentric. So if it is beneficial, then only we let it flourish, then only we let it survive, right? So physics and chemistry are also natural sciences. Biology, physics, chemistry and natural sciences. In biology, we'll study about plants and animals. Physics and chemistry gave rise to engineering, they gave rise to industries, and they gave rise to technologies. But this physics and chemistry knowledge also is used to human comfort and welfare only. They are also working on developing instruments, developing techniques which are helpful for the human comfort and welfare only. So the major thing what we need is the food, right? So the major thing what we get from the nature is the food. So the major utility of the biological world is the source of the food. Now coming to that point, we can tell biotechnology. Biotechnology, it's an offshoot of 20th century modern biology. Biotechnology is not a natural science, it's an applied science, right? It's an offshoot, means it's a branch from the main natural science. When did it came? It came in the 20th century. Biotechnology, which is the 20th century offshoot of modern biotechnology, modern biology, changed our daily lives. Whatever products we are getting from biotechnology, you know, so they have changed our lives, they have added comforts, they have added values, right? So they changed our lives because the products brought quantitative improvement in our health. Normal vaccine and biotechnological vaccine. rDNA vaccine has so much of difference in that rDNA vaccine is much safer, right? And it's having more efficacy than the normal vaccine. So the biotechnology products has improved our health and improved the food production also. We will see how biotechnology is in food production also, right? So this is a historical account. Now you are going to check the NCERT textbook and tell me, like, where is this information? In NCERT textbook, where is this information? Check it and post me in the comment box. Okay, so in this manner we have to read NCERT. Whether there's a historical account, that also we need to take into account. Whether there is a scientific discoveries, that also we should study. Not only the content, we need to study this information also. Now let's talk about two scientists, Cohen and Boyer. So we can tell they are the pillars behind the biotechnology industry. So we'll see like what works they did. So how did they build up the concept of biotechnology? How they laid the pillars for laying biotechnological factories and industries. Let us see. So first let us talk about Herbert Boyer. He is born in 1936. Boyer is born in 1936, where he is born in Western Pennsylvania, where making the railroads and all is a major work for the youngsters. From such a background, he came out, he studied and he completed his graduation. He completed graduation from the University of Pittsburgh in 63. He is born in 36. He completed his graduation in 63, where from the Pittsburgh, and 66 from Yale he completed his PG also, post-graduation also. In the same year, in 1966, he joined as an assistant professor in the University of California, San Francisco. He joined when 1966. Then in 1969, he started working on a research topic which is about restriction enzymes. So he told in 1969, he started working on REN means restriction endonucleases, restriction enzymes. He observed that there are certain enzymes which cut the DNA precisely in a required fashion. They cut and they leave sticky ends. They leave sticky ends. These sticky ends, which are called clip ends, can easily join. They can easily join to make a recombinant DNA. This is an observation made by Boyer. Is born in 36. He completed his graduation in 63 from the University of Pittsburgh. He completed his post-graduation in 66. In the same year, he joined as an assistant professor in the University of California, San Francisco. 69, he started working on couple of restriction enzymes extracted from a bacteria called E. coli. Remember, restriction enzymes you cannot manufacture in the lab, or we cannot prepare it. Only prokaryotic organisms will prepare those enzymes, right? So he worked on some restriction enzymes which are cutting the DNA in a precise manner, giving sticky ends, giving staggered ends, clip ends, which can be easily joined to make an rDNA. Now this was a great discovery. Now that discovery he was explaining to a scientist at Hawaii. Boyer is discussing his discovery at Hawaii with a Stanford scientist. What is his name? Stanley Cohen. Now he was telling all his discoveries to Cohen. Then at that time, what Cohen is working on? Cohen is working on small ringlets. He was working on extra chromosomal DNAs which are present again in the bacteria only. They are called plasmids. So Cohen is studying small rings of DNA which are called plasmids. They freely float in the cytoplasm of the bacterial cell, and they are called autonomous replicating elements because they replicate by themselves. So they are called autonomous replicating elements. They are extra chromosomal. They are not chromosomal. They are present in the cytoplasm and they replicate themselves from the coding strand of the DNA. So plasmid DNA replicates from the coding strand of the DNA. Now Cohen, what did he do? He developed a method to take out the plasmid from the bacteria. He developed a method to isolate the plasmid from the bacteria, and then he developed a method to insert it back into the bacteria also. You are isolating it out and then sending into the bacteria also. Now then, when Cohen has removed the plasmid out, Boyer's restriction enzymes are there, no? So those enzymes were able to digest the plasmid. They were able to digest the plasmid. When you digest the plasmid, then the plasmid will open, a circular plasmid will open. When it opens, your desired DNA, foreign DNA, alien DNA, so anything which you want to clone, so bring that DNA and keep it back here, then join it. And now this is not a simple plasmid DNA. A plasmid DNA is acting as a vector. Now it is carrying your desired DNA, and this modified plasmid is sent into a bacteria again. So Cohen know how to remove it, Boyer know how to cut it, and then how to modify it, and Cohen know how to send it back. So now these two people's discoveries together help to start the biotechnological company. So they prepared an rDNA, they sent it back into the bacteria. Now this bacteria acts as a manufacturing plant. The bacteria acts as a manufacturing plant to produce the proteins or to produce the genes, right? So we are using bacteria to produce the genes in n number of copies, which is called as gene cloning. So this is a foundation for biotechnological industry, right? So this is there in the starting of the chapter. This I told, check and tell me in the comment box where is this information there. So this is also important. Take a screenshot, then we will get into the starting of the topic. What is biotechnology? Right? Now I told you who coined the term biotechnology. Karl Erke coined the term biotechnology. And who is father of biotechnology? Paul Berg is father of biotechnology, right? So what is biotechnology? Biotechnology is an offshoot of 20th century modern biology. So the tools or the products of biotechnology are improving our comforts, are adding lives, they are safer, they have developed our health, right? All those things we discussed. Now biotechnology means applying technology to develop goods and services which are useful for mankind. So whatever which are useful for mankind, which is a biological organism, we can call it as biotechnology. So let's start. Biotechnology word was coined by, who coined the term? It was coined by Karl Erke. And who is considered as the father of biotechnology? So we are telling it is Paul Berg is considered as father of biotechnology. Now, if we have to discuss biotechnology, so biotechnology, it helps in the development of tools and techniques. It helps in development of goods and products which help for building up our welfare, which helps in building up our welfare. But then, if you see, you are telling biotechnology is an offshoot of 20th century, but then if you are using microorganisms for developing goods and services, which are useful for welfare. Making curd, making wine, making cheese is also biotechnology because you are using microorganisms. Cheese is good, curd is good, it is having nutritional values. All that is also biotechnology then. But if we have to restrict that traditional biotechnology and tell this is modern biotechnology, which is an offshoot of 20th century, so we can tell that if we are using the wild organism as such, then that is traditional aspect of biotechnology. If we are using a genetically modified organism for producing antibiotics or for producing vaccines or for producing hormones and all, so then it is called as modern biotechnology. So let us see what is traditional biotechnology. So we are telling traditional biotechnology means we use the microorganisms as such. We use the microorganisms as such to produce products and services. But when we take modern biotechnology, now examples, what are the applications of traditional biotechnology? Means curd production is one application. So we are taking normal bacteria, that's lactobacillus, which helps in curd production. Curd production we can take, and then we can go with alcohol production. In alcohol production also we are taking the microorganism as it is, we are not modifying it. Cheese production, all these are applications of traditional biotechnology. But when it comes to modern biotechnology, what do you mean by modern biotechnology? In modern biotechnology, we tell that we use the microorganisms, but they are not natural microorganisms, they are genetically modified organisms. So we use genetically modified organisms. When we use genetically modified organisms, then it comes under modern biotechnology. So in modern biotechnology, we can tell antibiotic production, we can tell, then we can talk about insulin production, hormone production, enzymes production, rDNA vaccine production. All this comes under modern applications of biotechnology. Now, not only all those things which are related to health sector, we can also tell that what are the other techniques which also comes under biotechnology, means the other processes or the techniques which comes under biotechnology. We can tell IVF-ET. What do you mean by IVF-ET? In vitro fertilization, embryo transfer, which we commonly call as test tube baby production. Test tube baby production, right? Is also application of biotechnology only. It's also application of biotechnology. Synthesizing an artificial gene is also a process of biotechnology. Developing DNA vaccine is also application of biotechnology. And gene therapy, correcting the defective gene, is also a process of biotechnology. Not only vaccine making, not only antibiotic production, so it also includes the process and techniques like IVF-ET we can tell. Then we can tell artificial synthesis of DNA. Artificial synthesis of gene also comes under biotechnology. Then correcting the defective gene is also application of biotechnology. Correcting the defective gene. Now correcting the defective gene means it comes under gene therapy. And production of rDNA vaccine is also a technique of biotechnology. Production of rDNA vaccine is also a technique of biotechnology only. Now, if we see, like, everyone are making their own definitions about biotechnology, then there is an organization which is called European Federation of Biotechnology. Now, they have given a proper definition of biotechnology, keeping the traditional view also and taking the modern molecular biotechnological view also. They have given a definition of biotechnology. So, what is that organization? It is called European Federation of Biotechnology. So, they have given the definition of biotechnology by taking this traditional view also, They have taken the traditional view also, and they have taken the modern molecular biotechnological view also, and they have given the definition of biotechnology. Let us see what is the definition of biotechnology given by EFB. So it tells that it is an integration. I told it's not a natural science, it is an offshoot. It's an integration of natural science, or sometimes we can take organisms, we take the cells, bacterial cells also, or if cell is not there, we take the part of the cell also. If that part is also not there, we can manufacture artificially. It's called molecular analog. So what is biotechnology? It's an integration of natural science, or we can take the cells, or we can take the organelles, or we can take part of it, or at least we can prepare the molecular analog, so to produce products and services for mankind. Let us write down the definition of biotechnology. So what are we telling? So it is the integration of natural science. It is the integration of natural science or organisms or cells or its products or its parts, we can tell. We can take cell or we can take part of it, or its parts, or at least molecular analogs or molecular analogs. We are using all these things for products and services of the mankind. So for using all these things, we are developing products and services for mankind. So this is a definition of biotechnology given by European Federation of Biotechnology. Take a screenshot, then we'll talk about the principles of biotechnology. Right? So when we have to tell what are the principles of biotechnology, each and every line and point of NCERT we are covering, children. In this manner only you have to read biology for NEET examination. Now when we talk about what are the principles of biotechnology, there are two core techniques they will tell. One is called genetic engineering, other one is called biochemical engineering. So let us see what are the principles of biotechnology. In that we will tell the two core techniques of biotechnology. What are the two core techniques? So in that one technique we call it as genetic engineering. And the other one is called chemical engineering or biochemical engineering or bioprocessing engineering. Now, what do we do in genetic engineering? What do we do in chemical engineering? Means whatever organism we have in which we want to modify the DNA, so that we do, right? So we will alter the chemistry of the genetic material. So in biotechnology, we will alter the chemistry of the genetic material. Let it be DNA or RNA. We will alter it, make it an rDNA, recombinant DNA, and then we will try to introduce it into the host organism, so that after taking that new DNA, for example, in bacteria we have kept our human genes, insulin gene. So then the bacteria suddenly started synthesizing insulin. That means the phenotype of the host will change, and then it will pass on to its progeny. So children, tell me, what is genetic engineering, which is a core technique of biotechnology? So we will alter the chemistry of the genetic material. Genetic engineering means altering the chemistry of the genetic material. Will alter the chemistry of the genetic material. In some organisms the genetic material is DNA, and in some organisms the genetic material is RNA. So we will alter it and we will introduce it into the host, right? And introduce it into the host. We will send it into the host and changing the phenotype of the host. Now the host gets changed because recombinant DNA came, and changing the phenotype of the host. It will change the phenotype of the host. So till here it comes under genetic engineering. So we took a bacteria, in that bacteria we have sent some new DNA in which rDNA is there, which is producing insulin or which is producing vaccine, whatever you want. So you can make a recombinant DNA, rDNA vaccine. So this recombinant DNA in which the bacteria is carrying, the bacteria phenotype got changed. This comes under genetic engineering. Now this bacteria should be multiplied. So with one bacteria your purpose will not be solved. You need to produce it in industrial scale. That comes under chemical engineering. So we tell that in chemical engineering we'll take a big bioreactor. In that big bioreactor we'll maintain sterile conditions, contamination free conditions, ambient conditions, and then we'll drop this bacteria. We'll provide required aeration, required agitation, proper food and all we will maintain, so that the bacteria keeps on multiplying by binary fission and it will produce your DNA to the large scale, so which will meet the demand of the population. So that comes under chemical engineering. What is chemical engineering? Taking a bioreactor or biovessel and carrying out fermentation. In that vessel we are going to maintain sterile conditions which will enable the growth of only the desired organisms. So in chemical engineering we will tell we may maintain sterile conditions. What do you mean by sterile conditions? Contamination free conditions. We'll maintain sterile conditions for the growth of only the desired bacteria will grow, for the growth of desired DNA. So desired DNA containing host only will grow, desired bacteria will grow, and then it will multiply and it will produce large scale desired growth of desired DNA and produce large quantities. It will produce large quantities by passing the DNA to the progeny. And by passing, how it will produce a large number? The bacteria will pass it to the progeny, passing the DNA to the progeny cells. Now that means I am telling, so here we made the bacteria, and this bacteria, when you provide proper nutrient conditions and all, then it will multiply and it will divide by binary fission. Now into the daughter cells also your DNA goes on. It is passing the desired DNA into the progeny. See, your DNA is going. Now again after some time this will divide by binary fission, and these daughter cells also will get the recombinant DNA. Again this bacteria also will divide by binary fission. That will also take the desired DNA. So like that. So we'll maintain conditions, sterile conditions, ambient conditions, contamination free conditions, so that the desired microorganism will grow. To which extent? To large extent, and it will manufacture the biotechnological products. What are the biotechnological products? It can be antibiotic, it can be vaccine, or sometimes it can be enzymes also. These two are the core techniques which gave birth to modern biotechnology. Take a screenshot, then we'll talk about what are the advantages of asexual reproduction, what are the advantages of sexual reproduction. So when we talk like asexual reproduction, there will not be any variations. That means asexual reproduction conserves the integrity as it is, whereas sexual reproduction will permit variations because meiosis is there during gametogenesis and synapses is there, crossing over is there. So we tell that, if we tell which is better means asexual reproduction or sexual reproduction, means sexual reproduction is advantageous or asexual reproduction. Why? Because it permits variations. So we'll tell sexual reproduction is better than asexual reproduction. Why we are telling sexual reproduction better than asexual reproduction? As it permits variations due to meiosis and all, variations will come. But when we talk about asexual reproduction will preserve the genetic material as it is, no variations, while asexual reproduction, what it will do? Will preserve the genetic material as it is. It will not change it. So there will not be any variations in asexual reproduction. There will be variation in sexual reproduction. So the nature won't change the organism so that it can better adapt to the environment. So that's why we tell sexual reproduction is advantageous than asexual reproduction. Now hybridization we do. What do you mean by hybridization? We'll take one color and we'll take another color and we do hybridization, so that we will get a mixed color. We'll get the characters of both these parents. So that is called traditional hybridization. Traditional hybridization, so when we do, we will get hybrid characters, but sometimes the hybrid characters will be desirable, sometimes the hybrid characters will be undesirable, right? So traditional hybridization technique allows multiplication. Traditional hybridization, excuse me, allows to produce hybrids with desirable combinations also. Sometimes desirable combinations will come, sometimes undesirable combinations also will come. With desirable combinations as well as undesirable combinations. But what do we want is only desirable combinations. We don't want undesirable combinations. So then to overcome this limitation, if we don't want undesirable ones, so that we tell to overcome this limitation, we are going to biotechnology. We are going to talk about genetic engineering, which is also called as gene cloning. Now, what do we do in gene cloning? So, we will isolate our desired DNA. This is our desired DNA. So, you have selected the desired DNA and we will try to insert this desired DNA into a recombinant vector. We will try to introduce this desired DNA into a recombinant vector. Since we are inserting into a recombinant vector, so there will not be any chance of getting unwanted characters. So then we will send it into the host cell. So then the host cell is carrying your desired characters only. So there is no possibility of getting undesirable characters, undesirable combinations. Isn't it beneficial? Isn't biotechnology beneficial when come to traditional hybridization? In traditional hybridization, beneficial characters also, unbeneficial characters also will come. But by using biotechnology, since we are selecting the desirable gene, we are keeping it into the vector, we are keeping it into the host cell, only desirable combinations will come. So that is application of biotechnology. So then the author has asked, what is the fate of alien DNA? When we take an alien DNA, what is alien DNA? Alien means which is foreign. See here, to this bacteria, this black colored DNA is alien DNA. So when you drop an alien DNA somehow into the bacterial cell, so this is a bacterial cell and this is a bacterial chromosome. Into the bacterial chromosome, if you drop and alien DNA in this manner, they're asking what is the fate of the alien DNA. So my answer here is the alien DNA will not be able to sustain inside. It cannot maintain inside, and when the bacteria is undergoing binary fission, when it is distributing its genetic material to the daughter cells, it will distribute only its DNA. It will not distribute the alien DNA. That means the alien DNA is not able to multiply, is not able to inherit into the progenies. Why it is not able to pass into the progenies means, so we will think, so if you, another way we will see, if we try to insert the alien DNA into the chromosome, this is a bacterial chromosome. Within the bacterial chromosome, if you now try to insert your alien DNA, then let us see what will happen. So bacterial chromosome, it has a site, a specific region called Ori site. Now with the help of Ori site, what happens? Bacterial DNA will replicate. Now when the bacterial DNA is replicating, along with the bacterial DNA, the foreign DNA, the alien DNA, what we gave will also get replicated, and you can see here it's being passed into the daughter cells. So that means if you give the alien DNA as such into a bacterial cell, then it fails to multiply it because it is not having Ori site. The other way, if you try to introduce into the bacterial chromosome which is having Ori site, then what happens? When the bacteria is dividing by binary fission, along with that your DNA also will multiply. When it becomes a part of the genetic material, then it will multiply, and this bacteria at Ori site, it starts its replication. Along with its DNA replication, our desired DNA also will get multiplied and it will be sent into their progenies, right? So that means here we can tell that ooryzite is very important for doing biotechnology. Take a screenshot, we'll continue. This is called gene cloning because your gene is getting cloned as many times as possible. Now later in NCERT, the next paragraph, they talk about who constructed the first rDNA molecule. Now I told you who are the people who act like pillars to build up the biotechnological industry. $1 million company industry means they're Cohen and Boyer, right? Cohen worked on plasmids, Boyer worked on restriction enzymes. So then obviously they will be the first people to construct the rDNA molecule. So our next topic is, when was the first rDNA molecule constructed? When was the first rDNA molecule constructed, or what was the first rDNA molecule constructed? So this point is very important. In NEET 20 also there was a question in this paragraph, and in NEET 2021 also they asked one question from this paragraph. So we can tell that in 1972, in the year 1972, Cohen and Boyer, they constructed the first rDNA molecule. They constructed the first rDNA molecule. Now, what is the alien DNA or foreign DNA for Cohen and Boyer? What is alien DNA? Alien DNA means it is our desired DNA. We can also call it as a foreign DNA for the host. Now, for the first time when they're constructing an artificial DNA molecule, the desired DNA for them is antibiotic resistant gene marker. What is an alien DNA for them? It is antibiotic resistant gene, right? So they have taken an antibiotic resistant gene. From where did they take the antibiotic resistant gene? From a bacteria called Salmonella. So from a bacteria, what is the name of the bacteria? Salmonella Panama. From this bacteria, they have taken antibiotic resistant gene which codes for kanamycin. So which antibiotic resistant gene it is? Kanamycin. From where did they isolate it? From Streptomyces. From Salmonella Panama they isolated it. Okay. Then this is your desired DNA. It should be kept into a vector molecule. Okay. So then from where are you isolating the vector? So they isolated vector from another bacteria. Now what is the other bacteria from which they have taken the plasmid? Means that bacteria from which they have taken these small ringlets. What is this? This is a vector. We call it as plasmid, right? Small ringlets observed by Cohen. Now this was isolated from Salmonella typhimurium. From Salmonella typhimurium, they removed the plasmid. Here is the plasmid. And from Salmonella Panama, they removed the antibiotic resistant gene marker, right? So now next step will be ligation. They have digested it and they have ligated using restriction enzymes. So first we will digest it with restriction enzyme. With restriction enzymes. So Cohen is a person who worked on restriction enzymes which cuts the DNA in a precise manner. We will see clearly about restriction enzymes. So if you want to insert this DNA into the vector DNA, then obviously you need to cut it. Restriction enzymes will cut the DNA in a specific manner. Since they are cutting the DNA, so we can also call restriction enzymes as molecular scissors. They are also called molecular scissors. Okay, now they have opened the plasmid, and then after opening the plasmid, we need to bring the desired DNA, kanamycin, and we need to join it. For joining, we need another enzyme which is called as DNA ligase. We used DNA ligase enzyme to join the vector DNA, plasmid DNA, with the alien DNA. So when you join the vector DNA with alien DNA, we get a recombinant DNA molecule. Now this particular one is recombinant DNA, which is also called chimeric DNA. It is also called chimeric DNA. Joining is done with the help of DNA ligase. So we can call DNA ligase as molecular glue then. What is it called? It is also called molecular gum, molecular glue. This is called molecular scissor, restriction enzyme, and DNA ligase is called as molecular glue. You joined it, we got recombinant DNA molecule. Now we need to send this recombinant DNA molecule into a bacterial cell, right? So then we need to send it into the host cell. They send it into the host cell. They have taken the desired DNA from from Salmonella Panama. They have taken the vector plasmid from Salmonella typhimurium, and now they are inserting into which host cell? Means they are inserting into E. coli, so which is having similar characters with Salmonella, Shigella, E. coli, Enterobacter. All of them, they come under a family called Enterobacteriaceae. They are sisters. They have similar characters. So that's why they were able to take it from Salmonella and they are able to send it into E. coli, and they want to see in E. coli whether this recombinant DNA is expressing or not. Yes, it is expressing. So they send the recombinant DNA into the E. coli, which is a close relative of Salmonella. So it went inside and it multiplied also. It went inside and it multiplied also. So that means if it is multiplying, it can pass to the daughter progeny. Your DNA is getting multiplied and is being passed to the daughter progeny. So this technology of multiplying n number of copies is called as gene cloning. We can call gene cloning, we can call it as rDNA technology, since rDNA molecule is getting cloned. So this is the first step of construction of rDNA molecule. In which year they constructed? 1972 constructed. Who constructed? Cohen and Boyer have constructed. What is the desired DNA? It is antibiotic resistant gene. Which antibiotic is it? Kanamycin. From where did they borrow it? From Salmonella Panama. From where did they take the vector? From Salmonella typhimurium. They have taken plasmid vector. They joined it, and into which host did they send it? In E. coli, which is able to multiply also. Take a screenshot, we will continue. Now this is construction of first rDNA molecule by Cohen and Boyer, and this technology is called as gene cloning technology. Now after that they have given what are the 3 basic steps for biotechnology. If we say biotechnology, altogether 7 steps will be there, but then what are the 3 main steps or basic steps? Means first one is isolation of desired DNA, right? And the second one will be sending the desired DNA into the host cell, and the third one is allowing the desired DNA to maintain in the host cell, and when the host is multiplying, it has to pass it into the progeny. So we can tell what are the 3 basic steps in creating a genetically modified organism. So what are the 3 basic steps in creating genetically modified organism? We'll see. What are the 3 basic steps in creating genetically modified organism? Means the first step is you have to fix what DNA you want to isolate. For example, if I have to produce human insulin, I need my desired DNA will be insulin DNA. Then I'll keep that insulin DNA in the bacteria. I'll ask the bacteria to multiply as many times as possible so that I can get so many copies of my insulin DNA, human insulin. It comes under biotechnology applications. So what is the first and foremost step? Means identification of the DNA with desirable genes. What's the first basic step? It is you need to identify identification of desired DNA. Identification of desired DNA is the first step. After identifying it, so we need to introduce that desired DNA into the host. That is the second step. Introducing the DNA into the host. We need to introduce the DNA into the host. Now, after introducing into the host, we need to do chemical engineering, bioprocessing engineering. So we need to check that we are providing all the ambient conditions for the growth of the host. Whenever the host is multiplying, then our DNA also gets multiplied. So the third step will be maintaining the introduced DNA in the host. Maintaining the desired or the introduced DNA in host and allowing it to pass into the progeny. we want to send the DNA into the progeny so that it can give as many copies as possible. So these are the three basic steps of creating a genetically modified organism. What is the first step? Fixing what is your desired DNA. Second one, introducing the DNA into the host. Third one, maintaining the DNA in the host cell such that whenever it multiplies, your desired DNA is passed into the progeny. Then the next technique is the tools of rDNA technology. What is the next step? So the tools of rDNA technology. So small r we need to write. Tools of rDNA technology. In rDNA technology, what and all we use, they come under the tools. Under that, the first one will be restriction enzymes. These are the main things. Then the second one will be DNA polymerase enzyme. And the third one, which joins, is called as DNA ligase enzyme. The enzyme which cuts the DNA is called restriction enzyme. The enzyme which will multiply the DNA, desired DNA, is called polymerase enzyme. The enzyme which will join to make a recombinant DNA is called DNA ligase. Then where are you joining your desired DNA? Into vector. So vector is the fourth rule. And where are you sending this rDNA? Into the host. Host is the fifth tool. So the basic tools of rDNA technology, first one is the enzyme which cuts, second one is the enzyme which multiplies, third one is the enzyme which joins, and the fourth one will be the vector, and the last one will be the host cell. Right? Take a screenshot, then we will talk about restriction enzymes in detail. Among the tools of rDNA technology, our restriction enzymes comes first. So I have taken all the information related to restriction enzymes on the board. We will see this and we'll continue. Now when we studied about Boyer and Cohen, also we studied that Boyer is the person who isolated restriction enzymes from E. coli, which makes sticky ends, which are widely used in genetic engineering. But then he worked in the year 1966. But in NCERT, under the side heading 11.1.1, they told that restriction enzymes were studied in the year 1963. That means there are some other people who studied restriction enzymes before Boyer. Now what are the names of those scientists? We will see here. Their names are Arbor, Smith and Nathans. Now what did Arbor do? What did Smith do? What did Nathans do? I wrote it on the board. Let us see. Now when Arbor is working on a bacterial cell, a restriction system of a bacterial cell, see, bacteria will be easily infected with a virus. The virus which is infecting bacteria is called bacteriophage. In order to safeguard its life from the viral attack, the bacteria will synthesize enzyme. The enzyme is called restriction enzyme, right? So only bacteria, prokaryotic organisms, can synthesize restriction enzymes. We or plants or other animals cannot synthesize restriction enzymes. Only bacteria will synthesize restriction enzymes. That too, why are they synthesizing restriction enzymes? Means to safeguard them against the viral attack, right? So here Arbor observed that. He observed that, like, he isolated two enzymes responsible for restricting the growth of the virus. Now two enzymes he identified. One enzyme is called methylase. The first enzyme is methylase. The second enzyme is restriction endonuclease. Now what will methylase do? Now the sequence, if it is methylated, then restriction enzyme cannot act on it. Now see, the bacteria has released a restriction enzyme. That restriction enzyme will check for a specific sequence and it will cut off that specific sequence. By mistake, the enzyme what the bacteria has secreted, if the similar sequence is there in the bacterial genome itself, then the enzyme will come and cleave off the bacterial genome and the bacteria will die, right? So that is why, to safeguard its DNA from the restriction enzyme action, first it will methylate the DNA. So by secreting an enzyme called methylase, the first enzyme, one enzyme adds methyl groups, that's called methylase. It adds methyl groups, and if the DNA is methylated, it is resistant to the action of nucleases. Nuclease cannot come and split it. In that manner, it will make its DNA safe. Then it will synthesize restriction enzyme. So it will be waiting for the virus. If the virus comes, then that restriction enzyme cleaves off the viral DNA. Understood, children? Now then comes next scientist called Smith. Smith observed what Arbor has told. Now Smith verified the Arbor's hypothesis and he isolated the first restriction enzyme enzyme from a bacteria called Haemophilus influenzae. Smith is a person who isolated a restriction enzyme from a bacteria called Haemophilus influenzae and he named it as HIND2. We will see the nomenclature later on. Let us finish off the discoveries. He isolated HIND2 restriction enzyme from bacteria called Haemophilus influenzae. He observed that this HIND2 enzyme will cut DNA in a specific manner. It will identify a specific sequence and it cuts that specific sequence in a specific manner. Now what is that specific sequence which is identified by HIND2? Means it is GTCGAC. The sequence what HIND2 is identifying to cut the DNA is GTCGAC. From 5' to 3' it is GTCGAC, and if you maintain the same orientation on a reverse strand, means again from 5' to 3' if you see, the other strand also will have the same sequence called GTCGAC. So it will identify such sequence and then it will cut it, right? That's why it's called nuclease. It cuts the DNA. Endonuclease cuts the DNA in between. Okay, now it is cutting between C and G. HIND2 is identifying a sequence called GTCGAC and it's cutting the bond between C and T on this strand, C and G on this strand, C and G on the opposite strand also. Means it is making cuts like this. Now if it is cutting such type of cuts, the result will be blunt ends. So it is cutting here and it is cutting here. If it cuts like this, it results in blunt ends. Blunt ends are also called flush ends. Flush ends means they are not having unpaired bases. They are having all paired bases. C is paired with G, A is paired with T, C is paired with G, G is paired with C, A is paired with T, C is paired with G. So all, if their sequences are paired, then that's called blunt ends, that's called flush ends. Means HIND2 is an enzyme which gives blunt end or flush ends. That's what Smith has observed. Coming to the scientist called Nathans, Nathans applied this restriction enzymes to create genetic maps, to create genomic maps. Okay, so Arbor identified that the bacteria will synthesize two enzymes to safeguard its life from the viral attack. The first enzyme is called methylase, which will methylate its DNA, making it DNA resistant to the action of nucleases, and the second enzyme is called restriction endonuclease, which specifically cuts the DNA in a precise manner. Smith verified it and he isolated the first restriction enzyme, which is called as HIND2, which reads a sequence called GTCGAC and which makes a cut between C and G, creating a blunt end fragments. Then Nathans used this restriction enzymes in genetics to construct genetic maps. For this, in the year 1978, Arbor, Smith and Nathans, they showed Nobel Prize. They shared Nobel Prize in medicine. In which year? 1978. Okay, so all this discovery was done in 63, right? Later in 66, if you see, Boyer is a person who isolated a couple of restriction enzymes from E. coli, so which will identify specific sequences and all. We studied in the starting of the chapter. Right now, today, how many enzymes are there available in the market? If a scientist wants to do genetic engineering, how many enzymes are available? Means 900 restriction enzymes are available in the market, isolated from 230 varieties of bacteria. 230 strain means varieties of bacteria. Okay, restriction enzymes, what are they? They belong to a large group of enzymes called nucleases. Nucleases are the enzymes which cut the DNA. So restriction enzymes belong to a large group of enzymes called nucleases. And if the nuclease cuts the DNA at the ends, if the nucleus is cutting the DNA at this end, if it's cutting the DNA at this end, so then that is called exonuclease. If it's exo means ends. If it cuts the DNA at the ends, it's called exonuclease. If the nucleus is cutting the DNA at the center, okay, in between, then it's called endonuclease, right? Restriction enzymes belong to a group of enzymes called nucleases. Now what these restriction enzymes are doing, so they are moving along the DNA, means they are inspecting the DNA. So they are in the identification of a specific sequence, which is called as palindromic sequence, which is called as recognition sequence. Now if you see me outside, you'll recognize me. In the same manner, here also, like restriction enzymes, they move on the DNA, they are inspecting the DNA and they are identifying certain sequences which are called as recognition sequences. Now HIND2 will recognize this particular sequence. In the same manner, each restriction enzyme will identify a particular sequence. It will identify identify a particular sequence, and what it will do, it will cut, right? What bond it is cutting? It is cutting the bond which is there between the backbone, which is there between sugar and phosphate, phosphodigester linkage it is cutting. So then it cuts the DNA, it cuts the backbone of the DNA, it breaks the sugar phosphate linkage. Sugar and phosphate are linked with phosphodigester linkage. It breaks that linkage and makes the DNA into fragments. So it can give a sticky ends, it can give blunt ends also. Now the recognition sequences of restriction enzymes. So this is a recognition sequence of a restriction enzyme called Hind2. The recognition sequences of restriction enzymes are palindromic sequences. Now let us try to understand what you mean by the word palindrome. So in NCERT textbook they have given Malayalam as a palindrome. Here I have taken the spelling of Malayalam. If you see M-A-M-A-L-A Malayalam, it should come in this manner. Yalam. Malayalam. 1 minute. Let me write it once again. If you had to write the sequence, Malayalam. Okay, so if you see here, it is Ma from this side, M-A, L-A from this side, L-A, Y-A, Y-A. So Malayalam is called as a palindromic word in English, which means the same in this manner, also in this manner also. Madam is also a palindrome. From this side also it is M-A-D-A-M. From this side also it is M-A-D-A-M, madam only. Literal is another example of a palindrome. Nitin, another name example of palindrome. So these are English palindromic words. Now in DNA, what do you mean by palindrome? So it seems that restriction enzyme will identify the palindromic sequences. In DNA, what do you mean by palindrome? Means, see, if you take GTC, GAC in the orientation of 5 to 3, then if you come the opposite strand in the same orientation 5 to 3, then it will be the same sequence it seems. GTC, GAC. Let us see GTC at the 5' end. In the back manner, if you see from the same orientation 5' end, it is GTC only. Here it is GAC, here also it is GAC. So palindromic sequences are identified by restriction enzymes, which are called as recognition sequences. They will cut them and then they will make fragments. Okay, so in DNA, a palindromic sequence means it is a base pair sequence which reads the same in both the strands when the orientation is the same. What do you mean by orientation? 5' to 3' orientation. Now here I have taken few palindromic sequences. We know GTC, GAC is a palindromic sequence for the first discovered restriction enzyme called Hind2, which is extracted from a bacteria called Haemophilus influenzae. And this is a popular restriction sequence, recognition sequence of an enzyme. It is an enzyme of a bacteria called E. coli. From E. coli, they have extracted this enzyme and it is called as EcoR1. The name of the restriction enzyme is called EcoR1. EcoR1's palindromic sequence or recognition sequences GAATTC. In the orientation 5 to 3. Come back in the same 5 to 3 orientation, it will be the same GAATTC. And this is a recognition sequence. Within the recognition sequence, here is a cleavage site where it cuts. Now here, where does it cuts? Means it cuts between G and A. It cuts between G and A. So it makes uneven cuts, but between the same two bases. Here also between G and A it's cutting. Here also between G and A it is cutting. So this is a palindromic sequence, means recognition sequence, and this is a cloning site or cleavage site for which enzyme? EcoR1. Now the second sequence is called GGATTC. Here 1G, 2 A's. Next time you write 2 G's, 1 A. GGATTC is a recognition sequence of another restriction enzyme called BAMH1. This is another restriction enzyme called BAMH1. So like we have names, your name is different, my name is different, other people names are different. In the same manner, there is another restriction enzyme called BAMH1. Its recognition sequences G 2 times it will come GGATTC. And where does BAMH1 cuts? Means BAMH1 cuts between G and G. It cuts between G and G. Means it is also giving sticky ends only. Now if you go to the third sequence, it is AAGE. So we are coming A 2 times and G 1 time. AAG and CTT is a sequence. This is a sequence of another restriction enzyme called HIND3. So this is a recognition sequence of another enzyme called HIND3. Okay. Now where does it cuts in the HIND3 sequence means? In HIND3 it cuts between A and A. And the last one, if you see, it is a recognition sequence for one more enzyme called PVU1. It is a recognition sequence of PVU1. Here also it cuts in between this sequence. So I showed some palindromic sequences with some restriction enzymes. Here we have seen HIND2. Here we are seeing EcoR1, one more restriction enzyme with its recognition sequence and cleavage site. The third enzyme is BAMH1. The fourth enzyme is HIND3, and the fifth enzyme is PVU1. These are the palindromic sequences and the restriction sites, cloning sites. Take a screenshot. We will continue. Right? Now we will see little more information about restriction enzymes. This is very important. Genetic engineering, gene cloning is possible with the discovery of restriction enzymes only. And Arbor, Smith and Nathan, we have to give credit to those people. That's why they got Nobel Prize. Since they have done a great discovery by isolating restriction enzymes from prokaryotic organisms. They will ask you, they asked once in NEET examination, what was the first restriction enzyme to be discovered? It is HIND2. And what is its recognition sequence? GTC, GAC. Is it a blunt end cutting enzyme or sticky end cutting enzyme? It is a blunt end cutting enzyme. So we saw the recognition sequences. They are palindromic sequences. So restriction enzymes cuts the DNA little away from the center. So REN means restriction enzymes, children. So restriction enzymes, they cut the DNA little away from the center. Little away from the center. But between the same bases on the opposite strands. But between the same bases on opposite strands. If we have to see an example, let us take an enzyme called EcoR1. It is a restriction enzyme. EcoR1's recognition sequence. What is the recognition sequence of EcoR1? It is 5' GAATTC. And if we have to see the palindromic sequence in the other manner, C pairs with G, T pairs with A, T, C, it is the same. Such sequences are called palindromic sequences. Within the palindromic sequence, where is a cleavage site? We told restriction enzymes will cut the DNA little away from the center. Center is here. Most of the cases, restriction enzymes will cut the DNA little away from the center. Here, the cleavage site is between G and A. It is between G and A. It's not at the center, slightly away from the center, but between the same two bases, between G and A only, between G and A only. This is the meaning of this sentence. Now, when it digests like this, when we have to draw it to show the fragments, then I need to show it in this manner. So, 5' G will be there and 3' C, T, A, because it has digested here. And the other fragment, if I have to see, it starts with A, T, C, 3' and this is 5' and here it will be G. Now these are sticky ends, means EcoR1 is an enzyme which cuts the DNA and it generated, these are called clipped ends. They are called clipped ends, or we can call them as sticky ends, or we can call them as staggered ends. They are called sticky ends or staggered ends. If they are sticky ends, if they are staggered ends, they are complementary to each other, and joining them becomes easy by hydrogen bonds. And what is it enzyme which joins these sticky ends? It is DNA ligase. The second enzyme in tools of biotechnology is DNA ligase. DNA ligase, it facilitates the joining of the clip ends easily. Facilitates the joining of the sticky ends. Easily it can join because they are complementary. AA can easily make hydrogen bond with TT. TT can easily make hydrogen bond with AA. Because of these sticky ends, DNA ligase can easily make hydrogen bonds and join it off. Understood? Now restriction enzymes made to construct rDNA molecule, right? So restriction enzymes, where are they widely used? In genetic engineering. For what purpose? To make recombinant DNA molecules. We can tell restriction enzymes are widely used. Where are they widely used? In genetic engineering. For what are we using them in genetic engineering? For making recombinant DNA. For making rDNA, recombinant DNA. I opened the fragment like this, and in between this fragment I can keep my DNA, alien DNA, desired DNA. See, I will do that. So I got some space here. In between this I'm keeping my DNA here, right? So which is having AATTTTA sequence. Now it is a heterologous DNA. It is having a black color DNA, it is having a red color DNA, then it's a recombination only, right? So our restriction enzymes will help to cut the DNA. By cutting the DNA, we can manipulate the DNA. We can keep whatever sequence we want in between, which makes the DNA to become recombinant. Take a screenshot. Then we will talk about the nomenclature of restriction enzymes. Right? Now, what do you mean by nomenclature? How to name a restriction enzyme? From where are restriction enzymes isolated? Only from prokaryotic organisms. Why are prokaryotic organisms synthesizing the restriction enzymes? To safeguard them from the attack of viruses. Now, nomenclature, we'll see enough. Let us take an enzyme which is there in NCERT for us. It is EcoR1. Now in this, if you see, this is the first alphabet, second alphabet, third alphabet, fourth alphabet, and there is a Roman number. 1 is a Roman number. Now let us see, what does the first alphabet represent? What does the second and the third alphabets represent? And what does the fourth alphabet represent? And what does the Roman number represent? Let us see. So in any restriction enzyme, you will be having a name like this, EcoR1. So the first and I told you, restriction enzymes are isolated only from bacteria. No other organism can make this restriction enzyme. The first letter denotes the bacterial genus. From which bacteria we isolated it? Its genus. For example, the first alphabet is E here. E denotes the genus. It denotes the genus. Now the genus is Escherichia. The genus is Escherichia. The second and third alphabets here are C and O. They denote the species. They denote the species. The species name is coli. There is a bacteria called Escherichia coli from which this enzyme is isolated. That's why its name is Eco. And the fourth alphabet is R. R denotes the strain. So the fourth alphabet R, it denotes the strain. Strain means variety. Okay, so it is RD strain. It is RD13 strain. Okay, so it's a variety. Like in mango, how many varieties are there? In rice, how many varieties are there? Here also, the variety of that E. coli is RD, resistant RD13 strain. And the Roman letter is there, right? Roman letter denotes its origin. Its origin means it's the first enzyme to be isolated from EcoR1. Next enzyme we isolate, it becomes EcoR2. So the Roman number denotes, it denotes the origin, or it is specific for that enzyme. Understood, children? So this is about EcoR1. Shall we try one more thing? So we know that the first restriction enzyme to be isolated is Hind2 by Smith, right? In that the first letter is H. H denotes the genus. What is the genus? It is Haemophilus. And I and N are the second and third alphabets. They denote the species. Now what is the species? Influenzae. Right? Genus name starts with a capital letter, species name starts with a small letter. So HI took capital letter because it's a genus name. Species name should start with a small letter. So I started with small letter. HIN, and D denotes the strain. It denotes the strain. RD, strain. And 2 is the order of isolation. Order of isolation. It's the second enzyme to be isolated. Okay. And what is the recognition sequence of Hind2? What is the recognition sequence of Hind2? We have seen that it is GTC, GAC, GTC, GAC, and it cuts between here and here, giving a blunt ends. And what is the recognition sequence of EcoR1? The recognition sequence is 5' GAATTC. So again GAATTC 3', and it cuts between G and A, giving a sticky ends. If you want few more enzymes in the vector, we will study in PBR 322 vector. We'll study there is a restriction enzyme called PVU1, which is extracted from a bacteria called Proteus vulgaris. It's extracted from bacteria called Proteus vulgaris. Okay, and one is the order of isolation. One more is there, PST1. PST1 tells that this is a restriction enzyme isolated from a bacteria called Providentia stroti. It's isolated from bacteria called Providentia stroti. Okay, then if we have to go with another restriction enzyme called BAMH1, BAMH1 is isolated from bacteria called Bacillus amyloliquefaciens. B stands for Bacillus, AM stands for amyloliquefaciens. Okay, then one more enzyme is there, SALVAN, is isolated from bacteria called Streptococcus albus. Streptomyces albus. Right, so PVU1, PST1, BAMH1, SALVAN, and then if we have to, these are some enzymes. EcoR1, Hind2 is there, Hind3 is there, Clavin is also there. It is from a bacteria called Caryophyllin latum. Clavin is isolated from a bacteria called Caryophyllin latum. So these are some enzymes, and this way the nomenclature goes. The first letter denotes the genus name, children. The second letter and third letter denotes the species name. If fourth letter is there, then it denotes the strain. In Clavin, fourth letter is not there. In Salvin also fourth letter is not there. In PVU1, PST1 also fourth letter is not there. If it is there, it denotes the strain, and then the Roman letter denotes denotes the order of isolation. And when we talk about restriction enzymes, how many types of restriction enzymes are there? Means type 1 restriction enzymes are there, type 2 and type 3 restriction enzymes are also there. Like we'll tell DNA Pol 1, Pol 2, Pol 3. Like that, in restriction enzymes also we have type 1 enzymes, type 2 enzymes and type 3 enzymes, but which are widely used in biotechnology, rDNA technology. Means type 2 restriction enzymes are only widely used because it can do in vitro amplification of the DNA. It can easily multiply the DNA outside also, and it cuts the DNA in a very precise manner. Up to the mark it cuts the DNA. Any extra sequences, extra burden it will not take. So that's why type 2 restriction enzymes are widely used. Take a screenshot, then we will talk about the fragments. Now we have digested the DNA. When we have digested the DNA, we are getting sticky ends. After getting these fragments, how will you separate them? There is a technique called gel electrophoresis, separation of DNA fragments. There is one side heading given in NCERT where they'll mention about gel electrophoresis technique, which will help to separate the fragments. Let's talk about gel electrophoresis. If we go with the NCERT order, so there is a technique which is called, under the side heading called separation of DNA fragments. When we digest the DNA with restriction enzymes, then we get fragments. Now we need to separate the fragments. What is the technique which is used for separating the fragments means the name of the technique for separation of DNA fragments is called gel electrophoresis. So the name of the technique is called gel electrophoresis. It was developed by a scientist called Tiselius. This technique was developed by a scientist called Tiselius. Okay. Now here, what acts as a solid matrix means the gel acts as a solid matrix. On the solid matrix, on the blackboard, on the whiteboard, I'm using to make you understand. I'm explaining the concept. In the same manner, if you have to separate the DNA, on what matrix are you separating the DNA? On agarose gel, we are separating. Gel acts like a matrix. And how are you separating means we are separating the DNA by subjecting it to electric field. So that's why electro word came. So electro refers to subjecting the gel and the DNA to electric field. And phoresis means gel will have pores. The gel will have pores. Through the pores, the DNA moves and it gets separated. Through the pores, DNA moves and it gets separated. Now, in the market, you will be having colored gel balls. Now, if you take two gel balls, between two gel balls, because of its circular shape, some space will be there. Through that space, the DNA moves on. Then that technique, when you subject it to electric field, it is called gel electrophoresis. Now, if you have to do gel electrophoresis, first we need to prepare the gel, right? So, then go and take a tray. So we need to take a tray. So I have taken a tray. In this tray, we need to take agarose gel. We need to pour agarose gel. Now agarose gel is extracted from the cell wall of red algae called Gelidium and Gracilaria. So agarose gel, from where do we get it? We extract it from the cell wall. From the cell wall of red algae, Gelidium and Gracilaria, we extract agarose gel and we mix it with the medium. We mix it with the buffer solution and we heat it. So gel, when it gets heated, it becomes that sticky, right? So take a tray and pour the gel in this tray. And when you're pouring the gel in a tray, so we need to take a comb. We need to take a comb. We comb our hair. So where teeth will be there on the comb. In the same manner, keep a comb and insert the comb in this manner so that wherever my fingers are there in the gel, they'll dip and they'll create spaces which are called as wells. In that manner, on this gel, we need to create wells here. So, I'm creating one well, second well, third well and four wells we are creating. So well, how do we create? By keeping a comb, we create a well. Now afterwards, when the gel, you have to cool the setup. When you cool the setup, what happens? The gel solidifies. After that, remove the comb. When you remove the comb, wells are there. So what are these called? So 1, 2, 3, 4. These are called wells. This is well A, B, C, D. Okay. So in these wells, we can load our DNA samples. In these wells, we can load our DNA samples. In NCERT, they tell that, so they have given ABCD instead of 1. They have given 1, 2, 3, 4. In NCERT, they mentioned that in well A, in well 1, so undigested DNA is kept. Undigested DNA, which is not digested by restriction enzymes. As such, the whole DNA is there, that is kept. Whereas in well 2, 3 and 4, the well 2, 3, 4 contains digested DNAs. Digested DNAs means you will get fragments. restriction enzyme has digested, right, we'll get fragments. So they told that, so in one well, they want to show us whether the process of digestion has happened or not. If it doesn't happen, how can we see the band? If the process of digestion is done, how can we see the band? So undigested DNA, they dropped in well 1, 2, 3, 4, they dropped the digested DNAs, and then they took this whole setup and they have kept it in a chamber where electric field is there. So here they kept one electrode called cathode, and the other side they kept another electrode called anode. They kept electrodes here, and through the wires they have connected it to a circuit. They made a circuit between the wells, between the two electrodes. Now what happens when we supply current from cathode to anode, and where is the DNA loaded? The DNA is loaded at cathode, no? It has been loaded at cathode. And in electric field, charged particles will move. DNA, is it having charge? Absolutely, DNA has charge, because DNA is a nucleic acid. Acid has phosphate group here, right? And the phosphate group gives negative charge. So DNA is loaded at cathode, and DNA has, deoxyribonucleic acid has phosphate group, means it has negative charge. Since it is having negative charge, because of that negative charge, it will be moving towards anode, right? So DNA, because of its negative charge, it moves towards anode. It will move towards anode when you subject it to electric field. When you subject it to electric field, it moves towards anode. Now it is moving towards anode. If there is a lean person in running race, they'll run fast. If it's a fat person, then they can't run fast, they run slowly. In the same manner, here also what happens, if the fragment is lengthy, 100 kilobase pairs fragment, no, like then it takes some time to move towards anode. If it's a 10 base pairs fragment or 20 base pairs fragment, it is small, right? It runs fast. So then you can see that the smaller fragments will move fast. This is a smaller fragment. Okay, the smaller fragments move fast, whereas the heavier fragments will take some time to move. The heavier fragments will take some time to move, and you can see the smaller fragments have moved fast, whereas since we have taken 2, 3, 4 wells digested DNA, so we got so many fragments. We told that in lane 1 undigested DNA is there, it's not been acted by restriction enzyme, that means the DNA will be as it is. Then you will get only one band, right? So this much fat DNA is there. This much fat DNA is there, right? So in this manner, DNA gets separated. Why it is getting separated? Because it is having a negative charge. Basing on the negative charge, when you charged particles move in electric field. But here, the separation is basing on its size only. Smaller fragments move faster towards the anode. Bigger fragments move slowly. So separation is on the basis of came in NEET 2016 paper, I think. So in Electrophoresis, DNA fragments are separated on the basis of size. They are separated on the basis of their size. Means we are telling smaller fragments move fast. Smaller fragments move fast and they reach the anode fast, whereas the bigger fragments, they move very slowly. And if we talk about DNA, it is a transparent particle, it doesn't have any color. Gel also doesn't have any color. Then how can you visualize the DNA? How can we visualize the DNA? That is a question here. Now we have some stains, we have some dyes. So there is a dye which specifically taken up by DNA only, that is called Ethereum bromide. So if you want to see the DNA bands like this, then you need to stain the gel with a stain. What is that called? The stain is called, or the dye is called Ethereum bromide. Ethereum bromide, what is it? It is a dye which stains the DNA sample. But Ethereum bromide is a fluorescent stain. It is a fluorescent stain. Fluorescent stain means it is seen only under UV light. It glows under only UV light. So that means this gel should be taken to a UV chamber, UV box, and there we'll switch on the UV light. When we switch on the UV light, we can see that in the presence of UV light, so the DNA which has taken the Ethereum bromide gives bright orange colored bands. So that's why I have used this color here to represent bright orange color. Gel is transparent. On the transparent gel, the DNA which gets separated, when we keep it under UV light, it appears as bright orange color bands. are seen under UV light. After we visualize them, after we locate them, then we need to extract it. We need to cut the bands, right? We need to cut the bands from the well. So cutting and extracting them. See, we are cutting them from and extracting them from the gel. This technique is called as elution. This also came in NEET examination. What is elution? Elution is cutting the DNA and extracting the DNA from the gel. Elution is cutting the DNA and extracting it out from the gel is called elution. So all this is about gel electrophoresis. Now when you digest the DNA with restriction enzymes, then what happens? We'll get fragments. Those fragments need to be separated. On what basis are you separating them? On the basis of their size. Smaller fragments move fast, bigger fragments will be at the cathode only. What is the technique called? Gel electrophoresis. Who developed this technique? Tiselius developed this technique. What is a solid matrix here? Agarose gel. From where did you get this agarose gel? From the cell walls of red algae called Gelerium and Gracilaria. How will you get these wells here? We'll get the wells by inserting a comb and then allowing it to cool it and solidify it. Now what is that wells meant for? In those wells we will load our DNA samples. Undigested DNA in one well we can load, and digested DNA to see the process we can load in the other things. After getting separated, we need to stain with Ethereum bromide, which is a fluorescent stain. So you need to check it under UV light only. Then you will find bright orange color bands, and then we will elute our DNA. Take a screenshot, then we'll talk about the steps of genetic engineering or the steps of biotechnology. Right, now let us see what are the basic steps of biotechnology. As I told in the starting of the chapter, there are 6 or 7 steps in biotechnology, but in the textbook, actually NCERT textbook, they did not follow an order. So first step is mentioned somewhere later, second step comes in between, and last step also comes in between. In that manner they have given it. So to get an idea, so we'll write down all the steps of biotechnology so that we'll understand what comes next. So steps of rDNA technology or biotechnology, if we go. The first one is you need to decide. The first point is you need to decide what DNA you want to isolate. So what is your alien DNA? What is your desired DNA? So isolation of the desired DNA is the first point. So what is the first point? The first point is isolation of desired DNA. So desired DNA is called as foreign DNA. You can also call it as alien DNA. We can also call it as gene of interest. So we are interested to multiply this gene. We want to clone this gene and get multiple number of copies. So that's why you can call it as gene of interest. We are taking it from other source, so it's called alien DNA. And alien and foreign is the same. And it's called desired DNA because we are interested in this. So suppose for example, you can take insulin DNA as a desired DNA. You can take insulin DNA as a desired DNA. This is your first step, isolating the desired DNA. Then your next step will be selection of the vector. What is the second step? Second step will be selection of vector. Now what do you mean by vector? Vector is a carrier. Vector is a carrier. It is a vehicle. Now malarial parasite Plasmodium, it's carried by mosquito. Mosquito acts as a vector, right? For example, from here if I have to go to my college, like bike is my vector. Bike will take me from my home to the college. In the same manner here, selection of a vector or a carrier which takes the desired DNA till the host cell. Okay, now selection of vector depends upon which host you are sending into. So selection of vector depends upon the host. For example, if we think if the host is bacteria, then what is the plasmid we have? What is the vector we have choice? Or else if the host is plant cell, then which vector do we choose? And if the host is animal cell, then which vector do we choose? If the host is bacteria, if you want to send anything into the bacteria, now who acts as a vector? Means plasmid acts as a vector. Viruses also can easily carry the gene into the bacterial cell, bacteriophages, right? So if the host is bacteria, the vector can be either plasmid or it can be a bacteriophage which infects bacteria. And if it is a plant cell, then the vector is Ti Plasmid. If it is a plant cell, then the vector we call it as Ti Plasmid. From where do we get this Ti Plasmid? We get this plasmid from a bacteria, and the name of the bacteria is called Agrobacterium tumefaciens. It is called Agrobacterium tumefaciens. So we will discuss about this in detail later on. Now, if bacteria is a host, then plasmid or bacteriophage acts as vector to carry the DNA into the bacterial cell. If plant is a host, then Ti plasmid from an Agrobacterium tumefaciens acts to transfer the DNA till the plant cell. If animal is a host, then retroviruses will act as vectors. If animal is a host, then who will act as vectors? Retroviruses. Adenoviruses will act as vectors, right? So this is a second point, selection of vector. Now the third step will be digesting the desired DNA also and the vector DNA also with the same restriction enzyme. So then the third step will be restriction digestion. Restriction digestion of desired DNA and vector DNA with same restriction enzyme, then only you will get complementary sites. We need to digest the desired DNA also and the vector DNA also with the same restriction enzyme. In NCERT textbook they showed us. So this is, if you take it as a desired DNA, in this desired DNA, if the sequence is GAATTC, opposite also it will be GAATTC only. Take it as a desired DNA. And if we go with vector DNA, in the vector DNA also the same sequence should be there. The same sequence should be there. It is GAATTC, GAATTC, and this is the vector DNA. So we are telling in the third step, the vector DNA and the desired DNA are digested with the same restriction enzyme. If you recollect, GAATTC is a restriction site or the recognition site for an enzyme called EcoR1. So I am bringing that enzyme here. The name of the restriction enzyme is EcoR1, whose palindromic sequence is GAATTC, who will cut between G and A. So then if we cut it with EcoR1, then what happens? G will be left out and C, T, T, A, A will be left out here. And this side, if you see, here it will cut here it will cut between G, A, A, T, T, C. Here it will cut and here it will cut. Right here the sequence will be G, C, T, T, A, A, and in this manner you will get vector DNA. So are they complementary to each other? T, T, A, A, T, T. Now the next step will be, we will join them. So we can do gel electrophoresis. Fourth step is separation of DNA fragments. Fourth step is separation of DNA fragments. Now just now we saw how to separate the DNA fragments. We will separate the DNA fragments by a technique called gel electrophoresis. So we will separate the DNA fragments by gel electrophoresis. After separating them, we can join them by an enzyme called DNA ligase. DNA ligase can easily join them, right? So next step will be DNA ligase action. When we join this desired DNA with the vector DNA, when we join this with the desired DNA and the vector DNA, then two DNAs are combining, then such DNA is called as recombinant DNA. DNA ligase, it helps in making recombinant DNA, rDNA we will tell. It is called recombinant DNA. So since recombinant DNA is made, we call it as rDNA technology. It is also called chimeric DNA. Chimeric means DNA from two different sources. It is called chimeric DNA. The first step is selection of the desired DNA. The second one is selection of the vector. Choice of the vector depends upon which host are we going into. And the third one is restriction digestion of the desired DNA and the vector DNA with the restriction enzyme. With the same restriction enzyme is important. Then after that we get the fragments. Those fragments need to be separated by gel electrophoresis. After separating them, we need to join them by using an enzyme called DNA ligase so that we get a recombinant DNA molecule. Now after getting recombinant DNA molecule, the next step will be sending rDNA into host. Sending rDNA into host. This recombinant DNA will be sent into host. Host is bacteria, host can be plant, host can be animal cell. We'll send it into the host. The technique is called transformation. Into the bacterial cell, the technique is called transformation. Into animal cells, it's called transfection. If you are sending the DNA into a prokaryotic cell, the technique is called transformation. If you are sending the rDNA into eukaryotic cell, then the technique is transfection. We will send it into the host cell. After sending it to the host cell, we need to check whether the host cell has taken the rDNA or it did not take the RNA. That means we need to do selection here. We need to do screening here to check for that bacteria in which rDNA is there. So, seventh step will be selection or screening. It will be selection or screening. So we screen and we take that host cell in which rDNA molecule is there. So after fixing, okay, this cell is having rDNA molecule, then we will go for chemical engineering, bioprocessing engineering, a big bioreactor. We'll add ample amount of medium. We will agitate and give aeration. We will check the pH. We'll check the temperature. All ambient conditions, favorable conditions, optimal conditions when you maintain, bacteria will multiply. Every 20 minutes it will multiply. Whenever it's multiplying, rDNA is also getting multiplying. That's called gene cloning. So the last step is called as fermentation. Fermentation. So in fermentation, in a big reactor, fermenter, or we call it as a bioreactor, we will grow the colonies. After growing that, at the end of the day, so we'll collect the product and we'll purify the product and we'll formulate it, which is called as downstream processing. So the ninth step is called as downstream processing. So all these side headings, you can find it in NCERT textbook, but they are not in an order, right children? So these are the steps of biotechnology. Now we have seen, we will now see isolation of desired DNA as a first step. Take a screenshot. Isolation of desired DNA in NCERT textbook, it's under topic 11.3.1. Isolation of desired DNA is under the topic 11.3.1. We can check there. Now, how can we isolate the desired DNA? Means the technique is called enzymatic digestion. Now, you have to break the cell, we have to break the cell wall, we have to break the cell membrane and the nuclear membrane, then get inside and take the DNA out. So, by using enzymes, we will break the DNA. So, that's why I'm calling isolation of desired DNA in NCERT. It's under topic 11.3.1. So, we will follow enzymatic digestion method. We will follow enzymatic digestion method. Suppose, for example, your desired DNA, if it is in a bacterial cell, then you need to break the bacterial cell wall. What is that enzyme which breaks the bacterial cell wall? Means lysozyme. You just add lysozyme, the cell wall will break and the DNA comes out. So in enzymatic digestion method, if the DNA is there in the bacterial cell, go with an enzyme lysozyme. And if the desired DNA is there in a plant cell wall is made up of cellulose, so then you can use an enzyme called cellulase to digest the cell wall. And if it is animal cell, no cell wall, right? So since cell wall is not there, so no need to digest it, it's not there. Then if it is in a fungal cell, if the desired DNA is in the fungal cell, we know that fungal cell is made up of chitin. So for digesting chitin, chitinase is an enzyme. So we digested the wall. Then for digesting the cell membranes, all cells will have cell membranes. For digesting the cell membranes, we need to add proteases and lipases. When we add proteases and lipases, cell membrane also will be digested and the nuclear membrane also will be digested. So in eukaryotic cells, if you see, now the DNA will be bound with histone proteins. It will be bound with histone proteins. So in order to remove these histone proteins, again we can use proteases. So what are we doing is ultimately removing all the fragments from the DNA, and then we are taking the DNA in pure form. So remember one point, when you need to isolate DNA, never use DNase enzyme. If you use DNase enzyme, your DNA will going to be break off, right? So that's why we will not use an enzyme called DNase. Instead, we will use proteases to break the protein. We'll use amylases to break carbohydrates and polysaccharides. We use lipases to break lipids. We use lysozyme, we use chitinases, cellulases like that, but not DNase. So then our DNA gets isolated out. Now this DNA which got isolated is there with all other unwanted components. You need to elute it out. Then we need to add chilled ethanol. When we add chilled ethanol, then the DNA gets precipitated out and it comes floating on the top. So after using all these enzymatic treatments, then how do you take the DNA out from that test tube, from that beaker? Means by adding chilled ethanol. If we add chilled ethanol, what happens means DNA gets precipitated out. This we can spool out. This is called spooling the DNA sample. When we take the DNA out, the technique is called spooling the DNA sample. So here we have done the first step, that is isolation of the desired DNA. Then after isolation of the desired DNA, what is our next step? Selection of the vector. Take a screenshot, we'll get into the second point, selection of the vector. Now selection of vector in NCERT, it's under topic 11.2.2 I think. So the second topic is selection of vector. In NCERT, it's under 11.2.2. As we already discussed, selection of vector depends upon the host. If bacteria is a vector, if bacteria is a host, then who can take the DNA to the bacterial cell is called as a vector. Now we know that Cohen has studied that the bacteria contains small ringlets of circular DNA which acts as vectors to carry the desired DNA into the host cell. So then the vector will be plasmid, or the viruses can easily infect the bacteria. The virus which infect the bacteria are called as bacteriophages. Now bacteriophages also can carry the DNA. Plasmid is double-stranded circular DNA like this. We can open the plasmid, and when you open the plasmid, you will get some space. In that space, you can keep your desired DNA. Then after keeping the desired DNA, send it into the bacterial cell, and the bacterial cell will take it because it is its DNA only, right? Plasmids means which are present in bacteria only. So it will take it easily, it will take it. Now in bacteriophase, what is bacteriophase? It's a virus which infects bacteria. In bacteriophase also it has linear double-stranded DNA. Now in its DNA also we will cut it, and how smart we are, see, we are keeping our DNA in between it. So when we keep our DNA here, then when the bacteriophage comes and infects the bacteria, so when the virus comes and infects the bacteria in this manner, when it comes and lands on the bacteria, so then what it will do? It will send its genome inside, right? So this is sending the plasmid. Here I am drawing it. And when the bacteriophage comes and infects, then also it will send the DNA inside. Along with its DNA, our DNA is also going inside. That means the bacteriophages are also carrying your desired DNA. The plasmid is also carrying your desired DNA. So that's why we tell plasmids and bacteriophages act as vectors if bacteria is a host. Now, so we'll take a plasmid and see what are the ideal characteristics that a plasmid should possess. So what are the ideal characters that a plasmid should possess? Now among plasmid and bacteriophage, which can carry bigger DNA inserts, means bacteriophage only can carry bigger DNA inserts. This is one advantage. And if the plasmid has to go inside, means bacteria only should take it, but without the permission of bacteria also virus can attack the bacteria. So means sending the DNA through bacteriophage is easy than the virus. So viruses can easily send the DNA. And since the viruses are very small in size, the number of how viruses DNAs will make more number of copies when compared to the plasmids. Means the copy number of the viral DNAs will be high. See how many times it has multiplied, how many copies we are getting. So we are getting so many copies. Means virus will give so many copies. Viral DNA will give so many copies. So these are the differences between plasmid DNA and bacteriophage DNA. Plasmid also is carrying the DNA, no doubt in that. Virus is also carrying the DNA, but virus can attack the bacteria very easily, and virus can carry lengthy DNA inserts. That's the second thing. And virus will make more number of copies inside the bacterial cell. This is the third advantage. Now when we have to talk about the why bacteriophage is able to make more number of copies, means the bacteriophage is having high copy number ability, whereas plasmids, I'm telling that bacteriophage has high copy number ability. Means what is my ability? I can take 3 hours class continuously. That's my ability. Someone can take 10 hours class continuously. That is their ability. So in the same manner, bacteriophage copy number is high. That's why it's making 1, 2, 3, 4, 5, 6, 7 copies it is making. Whereas if you go with plasmids, there are 2 types of plasmids. One plasmid per cell, it will be 1 or 2 plasmids per cell. If the number of plasmids are 1 or 2 per cell, that means the plasmid copy number is low. We call it as a low copy number plasmid. And sometimes the number of plasmids per cell will be 15 to 100 plasmids. If the number of plasmids are 100 to 15 to 100 per cell, then that is called as high copy number plasmid. It's called as high copy number plasmid. If I have given a choice for you, like which one you want to choose, a plasmid which is having low copy number or a plasmid which is having high copy number, obviously we want more number of our genes to be cloned. Then we will pick a plasmid which is having high copy number only. Understood? Now if we have to list down the characters what ideal plasmid should possess is, the first and foremost point what we write is called oricite. It is called oricite. What do you mean by oricite? This is a site which will start the process of replication. So in the same manner, when I'm starting the chapter also, we have seen introduction to Herbert, Cohen and Boyer we have seen. Then we have seen what are the applications of biotechnology, EFB, what definition they gave for biotechnology. That we came, and then we came into the process slowly. In the same manner, if your DNA should be, if your desired DNA should be cloned, if it should be multiplied, then it should be replicated. Replication starts with a sequence called oricite, right? What is oricite? It is the sequence where replication starts. It is the sequence where replication starts. If the oricite is strong, then it gives more number of copies. We will get more number of copies per cell. If the oricite is weak, it will give less number of copies only. Any DNA, if you keep with the oricite, for example, take this is oricite. Along with the oricite, if you keep your DNA, so since the replication starts at this point, along with this, your DNA is also getting replicated. So any DNA sent along with oricite, when we send along with oricite, will be multiplied, because oricite will help in multiplication. So oricite decides the strongness also. If the oricite is strong, more number of copies we get. And what's the significance of oricite? This is a place where DNA replication will start. This is a first point that a vector should possess. And the second point, take a screenshot. We'll talk about selectable markers then. The first character of a vector is it should have an oricite. The second character is it should have selectable markers. What are selectable markers? Let's try to understand. In most of the cases, the selectable markers are the genes which code for antibiotic resistance. Now for that, what is selectable marker? We need to take a vector called PBR322. So the first one is ORI site. A vector should have an ORI site, we are telling. And the second point, we should have, vector should have selectable markers. For understanding the selectable markers, let me draw a reconstructed vector or constructed vector which is called PBR 322. The name of this vector is PBR 322. So P stands for plasmid. It is a plasmid vector, and B and R stands for the scientists who constructed this plasmid. Their names are Bolivar and Rodriguez. And 322 is the order of isolation. 322nd time they were successful in building this plasmid. 320 times will we try? We don't have that much patience, we'll not try. But these are scientists, they have tried those many times to construct a plasmid, okay? So which is widely used plasmid. So PBR 322 as a vector should possess ORI site. That means it will be having an ORI site. Here is an ORI site of PBR 322. Then it should have two selectable markers, we are telling. The two selectable markers of PBR 322, one selectable marker is here and the other selectable marker is here. This selectable marker is called as ampicillin resistant marker, and the other one what PBR has is tetracycline resistant gene marker. PBR 322 has two gene markers as selectable markers. What are they? They are antibiotic resistant gene markers. They are antibiotic resistant gene markers. What are the antibiotic resistant gene markers which are present in PBR 322 means? One is ampicillin resistant gene marker, and the second one is tetracycline resistant gene marker. The other one is tetracycline resistant gene marker. So it has two antibiotic resistant gene markers, one is ampicillin, other one is tetracycline. We are telling that plasmid is having autonomous replicating capacity, means it doesn't depend upon the cell. So whenever it wants to multiply, it will multiply on its own. It will not wait for the bacteria to undergo binary fission. In binary fission, the genomic DNA will double, but whenever it wants, it will multiply. That is why a bacterial cell can have 15 to 100 copies of plasmids also. That means it has a specific gene. Here I am showing which is called as ROP gene. It has a gene which is called as ROP gene. ROP gene codes for the proteins which are required for the replication of the plasmid. So ROP, what does it stands for? ROP gene, it codes for the proteins. It codes for the proteins which are required for the replication of the plasmid. Whenever the plasmid wants to replicate, the ROP gene will make all the necessary proteins, and then the plasmid will replicate on its own. Then if we have to insert our desired DNA, we need to open the vector. Where are you opening the vector? So we will open the vector at the gene markers, at the selectable markers. Means you have a choice to open either at ampicillin or a tetracycline. I want to open at tetracycline. I want to open at that side. If I want to open at tetracycline, means I need to cut it, no? So if I need to cut it, so there should be restriction sites for restriction enzymes, recognition sites for restriction enzymes. Then what enzymes can digest ampicillin gene marker? What can digest tetracycline gene marker? Means if we have to see, ampicillin resistant gene marker has two cloning sites. One is called as PVU1, other one is PST1. In the same manner, tetracycline also has BAMH1 cloning site and SAL1 cloning site, and ROP gene by mistake PVU2 cloning site is there, and in between these two there is a region where three more enzyme sites are there. So one is called as CLAV1, Other one is called as EcoR1, and third one is called as Hind3. So how many restriction enzymes recognition sites are there? Means 1, 2, 3, 4, 5, 6, 7, 8. So which restriction enzymes are there in ampicillin? Means PVU1 and PST1. Which enzymes are there in tetracycline? Means BAMH1 and SAL1. Which are there in between these two? Means it is CLAV1, EcoR1 and Hind3 are there. Which restriction enzyme is there in DROP? Means PVU2. I told the nomenclature, how to name the enzymes. From which bacteria we have isolated it, we will take its name. For example, PVU stands for Proteus vulgaris. PST stands for Providentia strati. BAMH1 stands for Bacillus amyloleukophaciens. SAL1 stands for Streptomyces albus. CLAV1 stands for Caryophyllin latum. EcoR1 stands for Escherichia coli. And Hind stands for Haemophilus influenzae. Okay. So now this is a construction of TBR 322. In that, ampicillin and tetracycline acts as selectable markers. Now let us do that. So we are opening the plasmid at tetracycline resistant gene marker. So that means you can digest with BAMH1 and SAL1. In NCERT they told we are digesting with BAMH1. If you digest the vector with BAMH1, then tetracycline gets inactivated, it opens. Then I told you vector DNA also should be digested. The desired DNA also should be digested with the same BAMH1 only. This is a desired DNA. The desired DNA is also digested with BAMH1, then only complementary sites will be there. Then in the next step, we will add an enzyme called DNA ligase. When you add DNA ligase, then what happens? Into the vector DNA, the desired DNA will combine. So when the vector DNA combines with the desired DNA in this manner at the tetracycline place, then this is called as a recombinant DNA vector, rDNA vector. This rDNA vector, we did not touch ampicillin. Ampicillin is as it is. But what happened to tetracycline? Tetracycline got disturbed. Tetracycline resistance is lost. Tetracycline resistance is lost. Finally, you got recombinant DNA vector. So we took the desired DNA, we digested with BAMH1 restriction enzyme. We took the vector DNA, PBR, we digested this also with BAMH1. We opened the vector like this. We got the space. In that space, we kept the desired DNA. Then we used DNA ligase to join both of them. Since they are having complementary site sticky ends, you will get easily recombinant DNA. This recombinant DNA is sent into the bacteria. Now, sending the recombinant DNA into the bacteria is called as transformation. The technique is called as transformation. When you are sending into bacteria, then there is a possibility of getting 3 types of bacteria. In that, if you take this bacteria or we take this bacteria did not participate in transformation, means it is absent for the class in that manner. This did not participate in transformation, means call this as a non-transformed bacteria. It did not participate only, it's called non-transformed bacteria. If it is a non-transformed bacteria, then it will not have ampicillin resistance also, it will not have tetracycline resistance also. So then no, we can write in this manner, it is ampicillin sensitive, tetracycline sensitive. Whereas the other 2 bacteria are good bacteria, they participated in transformation. So we will call them as, this is transformed bacteria and this is also transformed bacteria. This is also transformed bacteria. So this is a transformed bacteria which has taken exactly this type of vector. Means it has taken the recombinant vector, it is lucky. So transformed bacteria with altered vector it is. This is transformed bacteria with the altered vector. We can tell it is recombinant vector. If it is a recombinant vector, then ampicillin is there, but tetracycline you disturbed it. In that place you brought your desired DNA. Then it will be ampicillin resistant, but it becomes tetracycline sensitive. It is resistant for ampicillin, but sensitive for tetracycline. Now come to the third bacteria. It is transformed bacteria. Sometimes what happens? You opened it. Meanwhile, you went to bring the desired DNA. Before the desired DNA comes in hurry, it will close by itself. They're complementary, it will close by itself. Then what happens? Then it's not a recombinant vector, it's not an altered vector, it's unaltered vector. So this, it has participating in transformation, but it took altered vector, it took unaltered vector with unaltered vector. So unaltered vector it took. Unaltered vector means as it is. Means ampicillin is there, tetracycline is there. Means it's recombinant or non-recombinant. It is non-recombinant. It is non-recombinant. If it is non-recombinant, then it has both. It is resistant for ampicillin also. It is resistant for tetracycline also. So now we are going for selection and screening. So when we are going for selection and screening, which type of bacteria do you want? You want to cultivate this bacteria where nothing is there? No, I don't want. Or you want to culture this bacteria in which recombinant DNA is not there? No. We want only this bacteria in which our desired DNA is there. Now how will you select it? For that we need to take a petri dish. We will take a petri dish. Inside this petri dish we will add nutrient agar medium. Along with nutrient agar medium, we will add the first antibiotic called ampicillin. When we add ampicillin in this medium, this bacteria which is ampicillin sensitive will die. This bacteria, it is ampicillin sensitive. When you give ampicillin, it will die. It cannot grow only. And again, this bacteria, it is ampicillin resistant. It will grow. So for our convenience, we'll take the numbers: first bacteria, second bacteria, and third bacteria. I'm telling that the first bacteria will not survive. The first bacteria, it will die. The second bacteria is ampicillin resistant, it will survive. The third bacteria is also ampicillin resistant, it will survive. So 2 survived and 3 also survived. Okay. Now what we will do? We'll consider this as a master plate. We will consider this as a master plate, and then we will prepare one more plate here which is called replica plate. We'll prepare one more plate called replica plate. We'll take a cheesecloth, we'll press it, and we'll take the impressions as it is. When you're taking impressions as it is, 1, 2, 3 at the same places, they start growing again. So 1, it's a dead bacteria only. 1 is a dead bacteria, so you can simply eliminate this bacteria also. 2 and 3 are there. Now this time in replica plate, don't add ampicillin, add tetracycline instead. Add the second antibiotic. What is that? Tetracycline, the second antibiotic we added. Now let's just check. The second bacteria, tetracycline, got disturbed because we kept our desired DNA. So that's why it became tetracycline sensitive. It will die when you give tetracycline. The third bacteria, where tetracycline is intact, it will survive. So that means here 2 is dead. Only 3 will survive. Now 2 is dead, 3 will survive. You want 3 or you want 2? I want that bacteria in which my DNA is there. Desired DNA, red color DNA is there. It is 2. So then we will check replica plate and master plate. Those bacteria which are absent in the replica plate, which are present in the master plate, we will pick those colonies from the master plate. Means I need to pick this bacteria which are not here, which are here. We will pick these colonies and we will go ahead. So this method is called selection. So how are you doing selection? How are you eliminating unwanted bacteria? How are you picking only the wanted bacteria, transformed bacteria, recombinant bacteria? Means with the help of selectable markers only, ampicillin resistant marker and tetracycline resistant gene marker. So a bacteria or a vector should have at least 2 gene markers. So this is one method which we have done selection by using PBr 322. But this method, we tell it is tedious because you are culturing them twice. Once on the master plate you are culturing, another time you are culturing them on the replica plate. So it becomes tedious. So they have introduced one more method. Take a screenshot. We'll talk about the next method called insertional inactivation method. There's another method which is easy, they're telling, which is with the help of another vector which is called PBr3, it's PUC vector. PBR stands for the scientist who constructed Bolivar and Rodriguez. Now what is PUC then? PUC is a vector which is prepared in University of California. UC stands for University of California, P stands for plasmid. Now let us see what are the gene markers which are present in the PUC vector. So 18, 19, a series of vectors are there. Now in this also we'll be having 2 gene markers. The first gene marker here is ampicillin resistant gene marker, antibiotic resistance, ampicillin resistant gene marker. The second gene marker is laxedzine, which codes for an enzyme. We'll study this in lagopron. Laxedzine, which codes for an enzyme called beta galactosidase. And as usual, it should have oricite here, and it should also have ROB gene here. So this is PUC vector. Now what we will do now is we will try to keep our DNA by disturbing the LAGZ gene. We have opened the vector. We disturbed the LAGZ gene. At the LAGZ gene, we kept our desired DNA, insulin DNA, we kept it. And you made an rDNA vector. Now this rDNA vector is sent into the bacteria by a technique called transformation. When you are sending into the bacteria, again what we have done earlier, you get 3 types of bacteria. This is one bacteria. This is a second bacteria and this is a third bacteria. Now second bacteria is transformed. The third bacteria, it took unaltered vector, and this is, it did not take, means we call it as non-transformed bacteria. It is non-transformed bacteria. If it is non-transformed bacteria, no plasmid only inside. Nothing is there inside. If nothing is there inside, can it show for amphicoelin resistance? No. So then tell it is amphicoelin sensitive. Is it having LAGZ gene? No again. So call it as LAGZ negative. Tell it is LAGZ negative. Whereas the second one, in which the desired DNA is there at the amphicoelin gene, it is transformed bacteria because plasmid is there. So call it as transformed bacteria. And the third one also, this is 1, this is 2, this is 3. This is also transformed bacteria. This is transformed bacteria with recombinant vector, rDNA vector. This is recombinant vector. This is transformed bacteria with non-recombinant vector. With non-recombinant vector. So if it is having recombinant vector, then amphicoelin gene, does it has? Yes, it has. So amphicoelin resistance, it is there. But when it comes to LAGZ, is not there. LAGZ negative it is. Come to the third one. Everything intact, it is there. A is also there. L is also there. That means it is amphicoelin resistant also. LAGZ positive also. It's amphicoelin resistant also. LAGZ also. Do you want the first bacteria or the second bacteria or the third bacteria? I want second bacteria in which the desired DNA is there, where I can take it and I can clone it. I don't want 1 and 3. So how will I do selection or screening? For that we need to take a Petri dish. We need to take a Petri dish. In that we are going to add nutrient agar medium. We are going to add nutrient agar medium. We also know that along with the nutrient agar, we'll add the antibiotic amphicoelin here. So we are adding the antibiotic called amphicoelin. And here we will add a substrate called X-galactose. We will add a substrate called X-galactose. All of us know that the LAGZ gene, it codes for an enzyme. It codes for an enzyme. The name of the enzyme is beta galactosidase. This enzyme breaks the disaccharides. It breaks the disaccharides and it gives monosaccharides. You will get colored product. You will get colored product if enzyme is there. Now here it is LAGZ negative. Here it is LAGZ negative, means it cannot make enzyme. Plasmid is only not there. Here also you disturbed the LAGZ gene and in that place we kept our desired DNA. So then it is also LAGZ negative, means this also cannot make beta galactosidase enzyme. The third bacteria, LAGZ positive. So nothing is there. It's having its LAGZ gene intact. Desired DNA is not there, means this one can produce an enzyme called beta galactosidase. It will produce an enzyme beta galactosidase. This beta galactosidase acts on X-galactose and converts the X-galactose and it gives colored colonies. It gives blue colored colonies, you know. So the third bacteria, it will give blue colored colonies. Why it is giving blue colored colonies? Because it is having intact LAGZ gene that will code for enzyme beta galactosidase that will break the X-galactose substrate to give chromogenic product. This is about the third one. Coming about to second bacteria, we're telling it is lac-Z negative. Gene is not there. It cannot make a beta galactose. It is enzyme. Then it cannot convert the X galactose into colored product. Then it will be white colored colonies. The second bacteria will be white colored colonies. The third bacteria are blue colored colonies. The first bacteria died, it didn't survive only. Which one you want? White colored ones or blue colored ones? I want the white colored one because in which my DNA is there, desired DNA is there. So pick it up. So pick the white colored colonies. So from one plate itself we picked. Isn't it easy when compared to that method, master plate, replica plate method? So this is called insertional inactivation. Inserting the desired DNA at the lac-Z gene has inactivated the gene. It has inactivated the lac-Z gene. So that's why this technique is called as insertional inactivation method. So I think you understood why I told that there should be at least 2 selectable markers in a vector. So one will help us to eliminate the transformants from non-transformants. The other one will help us to eliminate recombinants from non-recombinants. So 2 markers are required. Take a screenshot, then we will talk about the vectors. What are the vectors for plant cell? What are the vectors for animal cell? Now the fourth point under this characteristics of ideal vector. So we discussed about a vector plasmid, which is a vector for bacteria. If plant is a host, who is a vector? If animal is a host, who is a vector? That point also we'll discuss under point number 4. Now plants are easily infected by a bacteria called Agrobacterium tumefaciens. Plants are infected by a bacteria called Agrobacterium tumefaciens. This bacteria infects all the dicot plants. It infects all the dicot plants and it causes tumors to the plant. Why is this bacteria able to cause tumor? Means Agrobacterium tumefaciens has a plasmid which is called as Ti Plasmid. Agrobacterium tumefaciens has a plasmid. It is called Ti Plasmid. Ti, what does it stands for? Tumor inducing plasmid. It's called tumor inducing plasmid because it causes tumors. It is called tumor inducing plasmid. In tumor inducing plasmid, there's a black colored region which is called tDNA, tumor DNA. In Ti plasmid, it has a DNA called tDNA. What is tDNA? It is tumor DNA. So what will Agrobacterium tumefaciens bacteria do? Agrobacterium tumefaciens infects the plant, sends the Ti plasmid into the plant, sends the tDNA and causes the tumor in the plant. Now, why can't we use this to send our DNA into the plant? So what will we do? Now let's think on. So we have taken Agrobacterium tumefaciens. From Agrobacterium tumefaciens, we know how to isolate the Ti plasmid. Cohen has told us how to isolate the Ti plasmids. We isolated the Ti plasmid out. And this Ti plasmid, so we need to disarm. So my arm is my voice or my hand which I am explaining you. So if you remove my voice, I can't explain you. If you remove my hand, then also I can't explain you. In the same manner, the arm for this vector is tDNA, toxic DNA, tumor DNA. So disarm the plasmid. Because we don't want to cause the plant tumor, right? So it is harmful, it is dangerous. So that's why we are removing the tDNA. Removal of tDNA is called disarming the plasmid. What do you mean by disarming the plasmid? Disarming the plasmid means to remove the tDNA, the tumor causing DNA. When you remove the tumor causing DNA, I removed this tumor causing DNA. I got some space. In this space, yeah, we can insert our desired DNA. Our DNA, whatever is your DNA, disease resistant gene, go take it and go. So protein enrichment, we can do it. So then we disarmed the plasmid, and then in that place we kept our desired DNA. Then it becomes a recombinant vector. Send the recombinant vector into the bacteria. What's the name of the bacteria? Agrobacterium tumefaciens. It is Agrobacterium tumefaciens. And this Agrobacterium tumefaciens, now it's a natural genetic engineer, so it has an ability to infect the plant. So the bacteria will infect the plant. We use red color. The bacteria will infect the plant, and the bacteria will send your DNA. The bacteria will send your DNA. In earlier, what it is sending? Earlier it is sending the tumor DNA. Now it is sending our desired DNA. Now this plant got new DNA. This plant got new DNA. This is called transgenic plant then. This is called transgenic plant. So if you want to send anything into the plant cell, this is the best method we tell. What is it called? Agrobacterium mediated gene transfer technology. Agrobacterium, which is a soil bacteria. So it causes tumors in the dicot plants. It has a plasmid called Ti Plasmid. In Ti, tumor inducing plasmid, it has a DNA called tDNA. So tDNA causes tumor. So that's why we are removing that tumor part. We'll get space. In that space, we'll keep our DNA. We'll send it into the bacteria. Bacteria will send our DNA into the plant cell. Take a screenshot. We'll continue. In the same manner, if you want to send anything into an animal cell, the same method. Take adenoviruses or retroviruses. If you want to send into animal cells, then the vector is retroviruses. Now retroviruses also cause cancers in animals. They cause cancers in animals. Then do the same thing. From the retrovirus genome, remove the cancer causing gene. Remove the cancer causing gene. When we remove the cancer causing gene, we'll get space. In that space, keep your desired DNA. Means if you're taking agrobacterium also, if you're taking retrovirus also, we are doing disarming, right? We are removing the weapons. So in retrovirus also, we have to go with disarming the vector. We are removing the pathogenic part of it, and in that place we are keeping our desired DNA, and then we are sending into the animal cell or host cell. With this, we have seen the selection of the vector, right? So then what is the next topic? The first one is isolation of the desired DNA, that we have seen. Then selection of vector we have seen. Then next one is digestion, that we have seen. After digestion, separation we have seen. After that, selection and the screening also we have seen. After selection and screening, we need to take it and we need to go for bioprocessing. That we will see now. Take a screenshot. Now the next topic which we are going to discuss is PCR. Now let us see what is meant by PCR. So PCR stands for polymerase chain reaction. It stands for polymerase chain reaction. Now in this polymerase chain reaction, we can amplify the DNA, we can multiply the DNA. Now after cutting the DNA, we realized that the amount of DNA what we have got is very less. We want to make it little more before starting gene cloning experiment or rDNA technology experiment. Then we will use this technique called PCR. This technique was developed by an American biochemist. His name is Kary Mullis in the year 1970. So the technique of PCR was developed by an American biochemist. His name is Kary Mullis. In the year 1970s, he developed this technique. But it was an idea for this technique to develop. It took more than a decade. Okay, so the technique is called PCR. So this PCR, what is it? It comes under amplification of the DNA in a machine. What is the name of the machine where we are doing PCR? It's called thermal cycler. So the name of the machine is called thermal cycler, and it is an automated machine. It is an automated machine. It is called as thermal cycler. And who developed this? Kary Mullis developed it. What are we doing here? We are amplifying the DNA inside the plant body or animal body or bacterial body, or we are amplifying the DNA in a machine. We are amplifying outside the body. Means it comes under in vitro. In vitro means outside. Amplification technique. PCR comes under in vitro amplification technique, where we will amplify the DNA to how many times you want, we can amplify that those many times. The name of the machine is called thermal cycler. For doing PCR, so what is the processor means, we'll take it. Now let us take the DNA first. Let's take the DNA first. Here is the DNA, and these are the bases which are bounded by hydrogen bonds. Now this DNA in step 1 is subjected to denaturation. This DNA is subjected to denaturation. In denaturation, we will heat the DNA at 94 degrees centigrade for 5 seconds or 10 seconds. When we heat the DNA sample for some amount of time, what happens? The hydrogen bonds will get digested and both the strands get separated. So after that, after denaturation, we'll get our DNA sample like this, where the bonds are opened up, where the bases are opened up. Now in this step, after denaturation, double-stranded DNA got converted into single-stranded DNA. Only when you open the single-stranded DNA, you can read the bases A, T, G, C, A, T, G, C, like the sequences, the bases you can read. Now then, we need to start the process of replication by adding primers, complementary primers we need to add. So if this is 5 to 3, then the primer will bind here, because DNA replication happens in 5 to 3 prime direction. This is a complementary primer, and here it is 3 to 5, means the primer binds here in 5 to 3 direction. So what are primers? Primers are short stretches of DNA molecules which have complementary sequences to the DNA, and they bind to the DNA and they start the process of replication. So like this, only in NCERT they have given. So these are primer molecules. So the primer is going and binding to the complementary bases of the DNA. So this step will be annealing. So annealing will be step number 2. When you are denaturating, you are heating it to 94 degrees centigrade. When we are annealing, we will decrease the temperature, some 50 to 60 degrees temperature. When you decrease, then what happens? The DNA which is separated, the complementary bases are identified by these primer particles, and they will go and bind. When the primer binds, that means initiation of DNA replication started. It started the initiation of DNA replication. Means the daughter strand synthesis has started. Now after the daughter strand synthesis has started by the primers, then we need to continue it, right? For continuing, we need the building blocks, ATP, GTP, CTP, TTP, the building blocks we need. So we are going to add that, which together is called as DNTP mixture. DNTP mixture means deoxy ATP, deoxy GTP, deoxy CTP, and deoxy TTP. All these things we add. So they read, the DNA polymerase enzyme will read the bases which are present on the DNA template, and it keeps on adding the daughter strands. It keeps on. So it will add, and you have seen that this much daughter strand synthesis completed, and this much daughter strand synthesis completed. So this step is called polymerization. So the 3 steps of PCR are: first step is denaturation. In denaturation, when we heat, the double-stranded DNA becomes single-stranded. So we heat it at 94 degrees temperature. When we open both the strands in this manner, when we open both the strands, then the complementary primers will come and they will bind to the DNA molecules and they start the process of replication. After the replication starts, after annealing, then the third step, this step is called as extension. This step is called extension. Extension, we'll do it at 72 degrees centigrade. In extension, we will add the enzyme called DNA polymerase. We will add the enzyme called DNA polymerase, which will polymerize the daughter strand in 5 to 3 direction. Now at the end of the first cycle, we got 2 copies of DNA. This is 1 copy of DNA. This is the other copy of DNA. You started with 1 DNA molecules. At the end of first cycle, we ended with 2 DNA molecules. Again, they will go to the first step. Now this time 1 DNA separated, now 2 DNAs will separate, and the process will continue. So that's why I told this is a continuous cycle, polymerase chain reaction. It is like this. How many cycles you can continue? You can do up to 30 cycles. If you do 30 cycles, you will get a maximum of 1 million copies. 1 million DNA copies we get. Now this is the significance of PCR machine, which is developed by Carey Mullis. As I told you, it's an automated DNA sequencing machine. It's an automated DNA sequencing machine means, so like our washing machine, in which in one chamber we add surf, in other chamber we add conditioner, in other chamber we add starch, everything we add. The machine knows at which time it has to pick the surf, detergent, at which time it has to pick the conditioner, at which time it has to take the starch and all, it knows, and it will proceed. In the same manner here also, everything you'll add in the starting itself. You'll take small epind of a test tube type of structure in which you'll add the DNA sample. You're going to add the primer also. You're going to add the DNA polymerase also and the NTP mixture also, and activator metals like magnesium and all. DNA polymerase is activated by magnesium. That also we will add in the starting itself. In the first step, the machine will heat it to denature the DNA. In the second step, the temperature comes down. When the temperature comes down, complementary primers will go and bind. And in the third step, the temperature increases to 70 degrees centigrade, where tag DNA polymerase comes and it will continue the replication process. If we remember about DNA polymerase, enzymes are temperature sensitive. So they have very optimal range of temperature where they act. So here this enzyme has to sustain at 90 degrees temperature also, has to sustain at 60 degrees also, 70 degrees also. That means the polymerase which we are using in PCR, should it be a thermostable polymerase? If it is thermostable polymerase, then only it can sustain 90 degrees denaturation temperature. So that is why a special type of DNA polymerase we use here, we extract it from a thermophilic bacteria which is called Thermus aquaticus. This DNA polymerase is extracted from a bacteria called Thermus aquaticus. T stands for thermus, AQ stands for aquaticus. So TAC stands for Thermus aquaticus DNA polymerase, which can tolerate the temperature up to 100 degrees centigrade, because this bacteria is a thermophilic bacteria. It can survive at high temperatures. Now we are worried that our DNA sample is this much, so it can amplify and it can give you 1 million copies, 1 billion copies it can give you. So how nice the instrument is, it's amplifying and it's giving us this many number of copies. But the problem is it's very expensive. Everything you need to buy and add. Enzyme we need to buy and add. DNTPs we need to buy and add. Primers we need to buy and add. So it becomes very expensive. So after PCR, the next technique is chemical engineering, bioprocessing engineering. So we have done selection and screening, and we picked those bacteria in which recombinant DNA is there. That bacteria we want to culture now, we want to clone now, and we want to get as many number of bacteria as possible so that our DNA gets amplified. Now we do that in a bioreactor or a fermenter. Here in NCERT they have given us two pictures. This is one type of bioreactor and this is the other type of bioreactor where we are going to do chemical engineering, bioprocessing engineering. So bring that bacteria. So this is a bacterial suspension. We add the bacteria first. So we add the inoculum bacteria, we add, this is a lid, we add it here. And for culturing the bacteria, we need to provide food, that is called culture broth. We provided food, and we need to supply oxygen also. From the bottom, sterile air we are sending, and then water and food should be mixed with the bacteria. So then we need an impeller, we need a motor, we need an impeller rod which will mix it, which will mix the content properly. We can regulate the speed by this motor which is fitted on the top. So when we are mixing, what happens? Bacteria, medium and air, aeration, agitation will happen. So bacteria will take the air, they grow more, they multiply more. Inside your DNA is also getting multiplied. So this is an impeller rod, and these are the impeller blades which help in proper mixing of the content. And what type of culture medium do we add here? Means it is broth. Beef extract we add for culturing bacteria. Beef is rich in protein. Now if it is rich in protein, when we agitate it, foam gets generated. That foam becomes a problem. So we need to control foam here. How will we control foam? Means we have a foam breaking blades on the top. So foam has a tendency to come to the top. When the foam reaches the top, then these foam breaking blades will break them. But here blades are only a pair of blades. How can they break the entire foam? Don't think the compartment or the container is this small. It is 6-storied or 10-storied, that much big compartment will be there. In that much big compartment, that much wider compartment, a pair of foam breaking blades may not completely break the foam. And this foam, when it goes and touches the lid, cylindrical lid, dome shaped lid. Now the lid has so many openings. For impeller rod, one opening is there. For adding culture medium, one opening is there. For adding bacteria, one opening is there. For adding acid and base, one opening is there. For temperature thermometer, one opening is there. pH meter, one opening is there. Wherever opening is there, hole is there. through that hole, what happens, the foam when it's trying to escape, it goes and it breaks there and it brings the contaminated air inside. When the contaminated air comes inside because of the foam, then it disturbs the bacteria which is growing here. Contamination happened, unwanted bacteria is also growing, it is wasting the nutrients, it is wasting the space and all, so it's complicating the process. So somehow you have to control the foam here. So we can add anti-foam agent chemicals which break the foam. We can also use the foam breakers which can break the foam, or they have come with another technique we will see there which will control the foam there. So first we are talking about simple stirrer tank bioreactor. Bioreactor is a big vessel in which the bacteria will be cultured. So bacteria acts as an inoculum. For culturing the bacteria, we add culture broth here. For mixing the contents, we use the impeller rod and the impeller blades. We will pump the sterile air from the bottom and the motor will help in stirring, and we have a pH meter here. We can keep a pH meter here to check the pH. If the pH is going up or down, you adding acid and base, we can control the pH. So this is acid base reservoir. So if the pH is going high, add little bit of acid, pH comes down, right? If the pH is going down, add little base, pH goes up. And we also have a thermometer to check the temperature. This is thermostat which checks the temperature. This is pH stat which checks the pH. Now if the temperature is going high, how will you control the temperature? So the bioreactor is jacketed. It is having two walls. Between the walls, you can circulate cold air also, you can circulate steam also. When you circulate steam, temperature will increase. When you circulate cold water, temperature will decrease. So like that, we will regulate the temperature. By adding acid and base, we will regulate the pH, and then the microorganisms consuming the air, consuming the medium, they will grow nicely. And at the end of the day, when the nutrients gets exhausted, then what we will do, the bacteria starts entering stationary phase. They stop growing because nutrients gets exhausted. Then by the end of the day, we will open the tube at the bottom and we will collect all these things, and we will go for the last one, which is called as downstream processing, right? So this is about the first simple stirrer tank bioreactor, which is a cylindrical vessel. The only limitation here, the problem here is, what is a limitation? Foam is a limitation. Controlling foam becomes a problem here. When the foam is going up and touching the lid and it's breaking, it's bringing the contaminant air inside and damaging the bacteria with the unwanted things. Now here, if you see, that is called Sparger stirrer tank bioreactor. We should understand here what is Sparger. Sparger is a big plate-like structure which is having tiny nozzles, right? We can see here, this is a plate. Here, the plate is called Sparger. It is having tiny holes on it, nozzles on it. And what do we do means, we pump the sterile air. With high pressure, we pump the sterile air from the bottom through the tiny nozzles. So when you are pushing the air from the bottom, air has a tendency to go up. When the air is going up, now whatever the contents which are there on the top will come down. Air is going up, the contents will come down. Again, air will go up, contents will come down. That means here, aeration itself is acting as a mixing device, right? So then there's no need of those many blades and impellers. So I think one pair of impeller blades will meet the demand, are sufficient. Now and then occasionally if you rotate once, that is sufficient. Occasionally when you are rotating, then it might not generate that much amount of foam, which is going to be problematic. So the limitation here is a foam control. They have regulated here, and that, since it is having an extra part called Sparger, it's called Sparger. One pair of stirrer is also there. So Sparger stirrer tank bioreactor it is. Rest all, temperature control, pH control, jacket, all those things are the same with the stirrer tank bioreactor and the Sparger stirrer tank bioreactor. Now in this vessel, we will culture the bacteria, and by the end of the day, we will collect all the contents from the bottom, and we go to the last step, which is called as downstream processing. From the bottom, we are collecting. When do you collect this? When the biosynthetic phase is completed. When the bacteria has grown, when the biosynthetic phase is completed, then we will take all the things and we go with the series of steps. steps, right? We need to do purification, then we need to add formulations. So all these things before marketing or before packing, what we do comes under downstream processing. What are the steps which comes under downstream processing? Means separation of the compound, purification of the compound. So we can tell separation and purification are the main steps of downstream processing. After purifying it, so we need to add a preservative. So adding preservative is called formulation. So grade 1 preservative or grade 2 preservative or grade 3 preservatives we add it. Depending upon the preservative what we add, the life of the product depends. Whether it will expire in 3 months or it will expire in 6 months or it will expire in 1 year, like that it depends. First you separate the product, then you purify the product, and this separation and purification comes under downstream processing. Then go for formulations where we need to add the preservatives. After adding preservatives, we need to take this compound for clinical trials. What do you mean by clinical trials? Our product is tested on animals to check whether it is working or not, or any toxic chemicals got accumulated. So we can't directly test on humans, right? We cannot directly give it. So if anything happens, they can file a case on the company. So that's why every batch what they are conducting, so today morning to evening they have conducted this batch. So this batch, after preservative is added, it will be sent for clinical trials. After clinical trials, mostly stringently they'll do for drugs. After doing clinical trials, they have to go, it has to pass through the quality control tests also. QC department will be there in biotechnological company, which will take care of quality control and testing. They will check the quality is good or not, and they will see, like, is it for a human purpose, then how much purification it should be done. We can tell that the downstream processing and the quality control depends from product to product. If it is the same antibiotic for a veterinary purpose, quality level will vary. If it is the same antibiotic for the human level, so it will vary. So that's why the cost also varies. All these steps what we are talking comes under downstream processing. We will separate it, we will purify it, we will formulate it, then we will go for clinical trials, we go for quality control testing. After doing all these things, if it goes correct, then we will pack it and then we will go for marketing. Understood? All this is about the chapter Biotechnology, Principles and Processes. So here we have seen the PCR technique developed by Carey Mullis. Now this they have asked in NEET 2021 examination, they asked to arrange the steps of PCR in an order. The first step is denaturation, where the hydrogen bonds are broken by increasing the temperature. The second step is annealing, where complementary primers will go and start the process of replication. The third step is extension or polymerization, where tag DNA polymerase will go and will continue the replication process. By the end of one cycle, we will get 2 molecules of DNA. These 2 molecules of DNA, they go again, they repeat the process. You can repeat the process as much as 30 cycles, then you will be getting 1 billion copies of DNA. After this PCR technique, which comes under in vitro amplification technique developed by an American biochemist Carey Mullis, we came to bioprocessing. Under bioprocessing, we talked about the bioreactor. This is simple stirred tank bioreactor, and that's a Sparger bioreactor. Here foam becomes a problem. There that problem has been regulated. After doing this, from the bottom we'll collect and we'll go for downstream processing. Separation and purification are the main steps of downstream processing. After that we go for clinical trials. After that we go for quality control and testing. If you like the content, like, share, subscribe to my channel. So we'll meet soon with the next chapter. Thank you.